Hello there and welcome to Just About Earth, where curiosity gets cozy and science takes the long way around without hurrying anyone. Tonight, we are looking at a question that sounds simple at first and becomes more complicated the longer we sit with it. How long is it until the next mass extinction? It is not the sort of question that comes with a neat answer on a calendar. There is no official date circled in red, no countdown clock hidden in a laboratory, and no scientist standing beside a large button marked "Mass Extinction Begins Here." Nature is rarely that considerate. Large changes in life on Earth build through many smaller changes, some obvious, some difficult to measure, and some only understood after a great deal of time has passed. Before we get into any of that, where are you listening from tonight? Perhaps you are in a busy city where the loudest animal nearby is a delivery scooter. Perhaps you are somewhere quieter, with insects outside the window, birds settling into trees, or a fox making its rounds with the confidence of someone who has never paid rent. And while you think about that, consider one more question. Which living thing would you notice first if it disappeared from the place where you live? It might be a bird you hear every morning, a certain tree, a familiar insect, or even a creature you mostly complain about until it is suddenly gone. If you find this kind of calm, slow science useful at the end of the day, you can give the video a like and subscribe to the channel. It helps this small corner of the internet reach other people who enjoy learning without being shouted at by dramatic music every 12 seconds. There will be no flashing warning signs here, no urgent red arrows, and no thumbnail face looking alarmed at a picture of a dinosaur. We can take our time because understanding extinction properly requires more patience than panic. So settle in, get comfortable, and let us begin with the most important point. A mass extinction is not simply a period when many animals die. Death is part of every ecosystem. Species appear, spread, change, split into new species, and eventually disappear. That has happened for as long as life has existed. Most species that have ever lived are already extinct, and most of them vanished without a giant asteroid, a worldwide volcanic crisis, or a particularly bad week involving both. Scientists use the term background extinction for the ordinary rate at which species disappear over long stretches of time. It is not perfectly steady, and it is not easy to calculate because the fossil record is incomplete. Some organisms fossilize well, others leave almost no trace. A clam with a hard shell has a much better chance of appearing in the geological record than a soft-bodied worm that lived quietly in mud and never expected to become important to science. A mass extinction is different because extinction rates rise far above that normal background level and affect a large share of life across the planet. A commonly used benchmark is the loss of around 75% of species over a geologically short interval. That phrase, geologically short, needs a little attention. In everyday life, short might mean a few minutes. In geology, short can mean tens of thousands or even millions of years. Earth is very old, and it has a relaxed attitude towards scheduling. This is one reason the question, how long until the next mass extinction, is difficult. A mass extinction may not feel like a single event to the creatures living through it. It can be a long period of worsening conditions, repeated shocks, shrinking habitats, unstable climates, failing food webs, and populations becoming smaller and more isolated. There may be sudden disasters inside that period, but the full biological loss can take much longer to unfold. Imagine watching a forest over one afternoon. It might seem unchanged. Watch it for a year, and you notice seasons. Watch it for several decades, and you may see older trees vanish, new species arrive, rainfall patterns shift, and certain birds become less common. Watch it for centuries, and the entire system may be different. A mass extinction operates across many such systems at once, often over timescales longer than a human life. That makes it hard for us to recognize clearly, because our brains are much better at noticing a falling glass than a slowly emptying forest. There is also a problem with the word next. It suggests that mass extinctions are separate appointments, arriving one after another like buses. The history of life is not so tidy. Scientists identify 5 major mass extinctions in the fossil record, but there were also many smaller extinction events, regional collapses, and long periods of biological stress. The boundary between a severe biodiversity crisis and a true mass extinction is not always obvious while it is happening. The 5 major events are often called the Big Five. The name makes them sound like a group of unusually serious animals at a wildlife park, but they are the largest known extinction crises of the past 500 million years. Each had different causes and patterns. Some were connected to enormous volcanic eruptions. Some involved rapid climate change, changes in ocean chemistry, falling oxygen levels, or impacts from space. Often, several pressures work together. The most famous happened about 66 million years ago, when an asteroid struck near what is now the Yucatan Peninsula. It contributed to the extinction of the non-avian dinosaurs, along with many other forms of life. That event is easy to picture because it has a dramatic object at its centre. A huge rock arrives, the sky changes, food chains collapse, and the world becomes very unpleasant for anything with demanding dietary requirements. Other mass extinctions were less cinematic but even more severe. The largest known event at the end of the Permian period eliminated most marine species and a large share of land species. It was linked mainly to prolonged volcanic activity in what is now Siberia. Those eruptions released vast amounts of greenhouse gases, warmed the climate, altered the oceans, and helped create conditions that many organisms could not survive. There was no single afternoon when everything ended. The crisis developed through connected changes that spread through the planet's systems. That distinction matters now. When people hear mass extinction, they often imagine one giant catastrophe that has not happened yet. They picture an asteroid, a supervolcano, or perhaps something involving a very badly managed time machine. Then they look outside, see that the trees are still standing, and assume the danger must be far away. But a mass extinction does not need to begin with one spectacular event. It can grow from many pressures that reduce populations, remove habitats, and weaken ecosystems over time. If enough species disappear quickly enough, and if the losses spread widely enough, the result can meet the scientific definition even without a single moment that everyone recognizes as the beginning. This is where the question becomes uncomfortable. Many scientists have warned that current extinction rates are much higher than normal background rates. Habitat destruction, overexploitation, invasive species, pollution, disease, and climate change are all placing pressure on wildlife. These forces often act together. A species may survive a warmer climate if it can move to a cooler area, but not if roads, farms, and cities block the route. It may survive habitat loss if its remaining populations are healthy, but not if pollution has reduced reproduction. It may cope with one new predator, but not if it is already struggling to find food. None of this means that 3/4 of all species will disappear next Tuesday. It also does not mean that every species is declining. Some species adapt well to human environments. Rats, pigeons, crows, and certain insects have looked at our cities and decided that the arrangement is acceptable. A few have done extremely well, especially if they enjoy warm buildings, discarded food, and making noise near bedroom windows before sunrise. The broader concern is not that all life is vanishing at once. It is that many populations are shrinking, ranges are contracting, and ecosystems are becoming simpler. Extinction is the final step, but a species can lose most of its ecological role long before the last individual dies. A bird that once existed in millions may still survive in a few protected areas, yet no longer disperse seeds across a continent. A large predator may remain on paper while disappearing from most of its former habitat. Coral may persist in scattered places, while reefs lose the structure that supported thousands of other species. This creates something scientists sometimes call functional extinction. The species is not completely gone, but it is too rare to perform its previous role in the ecosystem. Think of a restaurant that still has one employee, one chair, and half a menu. Technically, it has not closed. In practical terms, dinner may be difficult. The loss of ecological function can spread. If a pollinator declines, plants that depend on it may reproduce less successfully. If those plants become scarce, animals that eat their fruits may also decline. If a predator disappears, prey populations may rise and change vegetation. Ecosystems are networks, not collections of independent species standing politely beside one another. Pull enough connections apart and the whole arrangement changes. Still, predicting when those changes become a mass extinction is extremely difficult. To do that, scientists would need to know how many species exist, how quickly they are disappearing, how future pressures will develop, and how ecosystems will respond. We do not have complete answers to any of those questions. Even the number of species on Earth is uncertain. Scientists have formally described roughly 2 million species, but the true total is believed to be much larger. Many insects, fungi, deep-sea organisms, microbes, and small tropical species have not been identified. Some may disappear before they are named. It is difficult to count losses from a library when many of the books have not yet been catalogued, and some are stored in rooms no one has entered. Extinction is also hard to confirm. A species can go unseen for years and then reappear. This has happened often enough that scientists are cautious. Declaring a species extinct too early can cause conservation work to stop, which would be especially unfortunate if a few individuals are still alive and waiting for someone to continue helping. On the other hand, waiting too long can make the official record lag far behind reality. There are also delays built into ecological decline. A species may be doomed by habitat loss but survive for decades because some adults remain. Long-lived trees can stand after they have stopped reproducing successfully. Large animals can persist in small populations even when genetic diversity is falling. These are sometimes described as extinction debts. The damage has already occurred, but the final loss arrives later. That makes a countdown almost impossible. If someone asks how long until a cake is ready, you can inspect the oven, check the temperature, and make a reasonable estimate. If someone asks how long until a global biological threshold is crossed, the oven is the size of a planet, the recipe is incomplete, and several people are changing the temperature while arguing about whether the cake exists. Scientists can still build models. They can compare present extinction rates with background rates, estimate future habitat loss, examine climate scenarios, and study how species respond to previous environmental change. These models do not produce one agreed date. They produce ranges, probabilities, and different outcomes based on what humans do next. That last part is essential. The future is not fixed. When someone asks how long until the next mass extinction, the answer depends partly on choices that have not yet been made. Rates of land clearing can rise or fall, protected areas can expand or shrink, fishing pressure can change, greenhouse gas emissions can increase, stabilize, or decline. Conservation programs can fail, or they can bring species back from the edge. There are real examples of recovery. Species once reduced to very small numbers have increased through legal protection, habitat restoration, captive breeding, reintroduction, and the removal of invasive predators. Whales have recovered in some regions after hunting restrictions. Birds of prey have returned after harmful pesticides were banned. Islands once emptied of native wildlife have been restored through careful, persistent work. None of these successes means the larger problem is solved, but they show that decline is not always irreversible. Mass extinction is therefore not a prophecy. It is a category scientists use to describe a scale and rate of loss. We can move closer to it or farther from it. We can increase the pressure on living systems or reduce it. The planet does not make moral judgments about this. Ecosystems respond to physical conditions. Forests do not care what was promised at a conference. Coral reefs do not read policy documents. Species respond to temperature, food, water, space, competition, disease, and whether enough members of their population survive to reproduce. That can sound cold, but it is also useful. Physical problems can be measured, causes can be identified, actions can be tested, a wetland can be restored, a migration route can be protected, a destructive fishing practice can be changed, an invasive predator can sometimes be removed, emissions can be reduced. Conservation does not require nature to understand our intentions. It requires us to change the conditions. The scale of the problem can make individual actions seem small. Turning off a light will not rescue every species. Planting one patch of native flowers will not stop global habitat loss. Refusing one plastic bag will not repair an ocean. But large environmental changes are built from laws, markets, infrastructure, farming systems, energy systems, public choices, and repeated local decisions. The important point is not to pretend that every small act has equal impact. It is to understand which actions affect the main causes and then support them consistently. There is another reason to avoid treating mass extinction as a distant finish line. If we wait until the scientific threshold is officially crossed, much of the damage will already be done. The difference between losing 10% of species and losing 75% is enormous, but 10% would still represent a severe and permanent loss. There is no sensible reason to ignore the first stages of a fire because the entire building has not burned down. The term mass extinction can also hide the uneven way loss happens. Some groups are more vulnerable than others. Species with small ranges, slow reproduction, specialized diets, or dependence on one habitat can be at greater risk. Island species often face serious danger because they evolved without certain predators and have nowhere else to move. Amphibians are highly sensitive to changes in moisture, temperature, habitat, and disease. Large mammals often need extensive territory and reproduce slowly. Reef-building corals are tied closely to temperature and ocean chemistry. Meanwhile, some hardy generalists spread. The result may not be a silent planet. It may be a more uniform one, where the same few adaptable species appear in many places while unique local species vanish. A city on one continent begins to share more of its common birds, weeds, rodents, and insects with a city on another. Biological variety is replaced by repetition. That is easier to overlook because life remains visible. There are still birds in the sky, insects around lights, and plants growing through cracks. Yet the composition has changed. A choir can keep making sound after most of its singers have left, but it will not be the same performance. The phrase "next mass extinction" also raises a deeper question. Are we waiting for one, or are we already in the early stages of one? Scientists do not all use the same wording. Some refer to the current period as the sixth mass extinction. Others say we are facing a biodiversity crisis that could develop into a mass extinction, but has not yet reached the scale of the Big Five. The disagreement is partly about definitions, timescales, and how current rates are estimated. It is not a disagreement about whether many species and ecosystems are under serious pressure. This difference in language can be confusing. If one scientist says the sixth mass extinction is underway, and another says it has not happened yet, it may sound as though one of them thinks everything is fine. Usually that is not the case. They may agree on the observed declines and disagree on whether the term mass extinction should be used before the full threshold is reached. Think of a doctor describing a patient with several dangerous symptoms. One doctor may say the patient is entering a medical crisis. Another may reserve that phrase for a later stage. They can disagree about the label while agreeing that treatment is needed now. The label matters for scientific precision, but the condition matters more. So, how long do we have? The most honest opening answer is that no one can give a precise number. Depending on future extinction rates, some estimates suggest that reaching the scale of past mass extinctions could take centuries or longer. Under worse scenarios, the process could accelerate. Under better scenarios, many losses could be prevented, and the threshold might never be reached because of human activity. The clock is not fixed, and it is not counting down at one steady speed. This uncertainty should not be confused with ignorance. We may not know the exact date, but we know a great deal about the direction of many trends. If a population is falling year after year, its habitat is shrinking, and the pressures causing the decline are increasing, uncertainty about the final extinction date is not reassuring. You do not need to know which minute a leaking boat will sink to understand that repairing the leak is a sensible idea. At the same time, fear alone is not a useful guide. Constant alarm can make people feel that the outcome is already decided. If the next mass extinction is presented as unavoidable, then conservation begins to look like rearranging furniture in a house that has already collapsed. The scientific picture is more demanding but also more hopeful. Many systems are under severe strain, some losses cannot be reversed, and the risks are growing, yet the total outcome remains open. Our task then is not to search for one dramatic date. It is to understand how mass extinctions happen, how the present compares with the past, which warning signs matter, and how quickly biological losses can build. We also need to look at what recovery means, because life has returned after every previous mass extinction. The recovery took millions of years and produced a world very different from the one before. That last point is easy to misunderstand. Earth will almost certainly continue to host life even after a severe extinction event. Bacteria are difficult to discourage. Insects are inventive. Small animals can survive in places that would make most of us reconsider our travel plans. The question is not whether all life will end. The question is what kind of living world remains, how much complexity is lost, and whether human societies can function well through the environmental changes that accompany that loss. Humans depend on ecosystems for food, water, soil, pollination, climate regulation, materials, and disease control. These services are not extra decorations added to civilization. They are part of its operating system. We can replace some natural functions with technology, but usually at great cost, and not everywhere. No one has built a machine that can replace all forests, oceans, soils, insects, fungi, and microorganisms while fitting neatly beside the washing machine. The next mass extinction, if it happens, will not be important only because many species disappear. It will matter because those species are connected to systems that support other species, including us. The risk is not that one morning every animal vanishes and humans stare sadly at empty gardens. The risk is a long sequence of losses and disruptions that make ecosystems less stable and human life more difficult. To judge how close that risk may be, we first need a baseline. We need to know what happened during the largest biological crises in Earth's past, how quickly they unfolded, what caused them, and what survived. The fossil record does not give us a perfect film of those events, but it gives enough evidence to show that mass extinction has happened in several very different ways. Some began in the oceans, some struck land more heavily, some were linked to sudden impacts, while others grew from climate and chemical changes that continued for thousands of years. Those earlier events are not exact templates for the present. There were no farms, factories, global shipping routes, power grids, or 8 billion people during the Permian extinction. Dinosaurs did not hold emergency meetings about atmospheric carbon. Trilobites did not issue reports on marine oxygen levels. Still, the past shows how quickly environmental pressures can spread through food webs and how long recovery can take once biological complexity is lost. It also shows that extinction is selective. Some organisms disappear while others survive and expand. Size, habitat, diet, reproduction, mobility, and tolerance for changing conditions all matter. Survival is not always about being strongest or most impressive. During a crisis, it may be more useful to be small, flexible, able to eat almost anything, and comfortable living underground. In other words, the future may belong less to the lion and more to the creature already hiding behind your kitchen cupboard. The 5 major extinctions each changed the direction of evolution. They removed dominant groups, opened ecological space, and allowed new forms of life to spread. Without the asteroid impact at the end of the Cretaceous, mammals might never have expanded in the way they did. Without earlier crises, the entire history of complex life would look different. Mass extinction destroys, but it also resets competition. The problem is that the reset takes place over millions of years, which is not especially helpful to anyone hoping to make plans for next weekend. Before asking what may happen next, then, it helps to look carefully at what happened before. The evidence is written in rock layers, fossil beds, changes in carbon chemistry, ancient lava fields, and the sudden disappearance of once common organisms. Each event left a different pattern, and together they show that the path toward mass extinction is rarely simple. It begins with pressure, spreads through connected systems, and reaches a point where ordinary recovery can no longer keep pace with loss. The first of those great crises began long before forests covered the land, when most complex life was still in the oceans, and the continents looked nothing like the map beside your desk. The world was colder, sea levels shifted, habitats vanished, and a large share of marine species disappeared. That ancient event gives us our first clear example of how a changing planet can reduce life on a global scale, and it is where the longer record of extinction truly begins. The first great extinction crisis in the fossil record took place about 444 million years ago, near the end of the Ordovician period. At that time, nearly all complex life lived in the oceans. There were no forests, no flowering plants, no birds, and no mammals standing on beaches wondering why the tide had gone out. The land was mostly bare rock, thin soils, and simple forms of vegetation. If you wanted to find the busiest parts of the planet, you had to look beneath the water. Those oceans contained communities of trilobites, brachiopods, corals, mollusks, and many other animals that would look unfamiliar today. Trilobites crawled across the seafloor in enormous variety. Brachiopods, which resemble clams at first glance but belong to a different group, covered many shallow marine habitats. Small floating organisms supported food webs above them. Life had spent millions of years becoming more diverse, especially in warm, shallow seas spread across the continents. Then the climate changed. A major cooling period began, and large ice sheets formed over the southern supercontinent of Gondwana. As water became locked in ice, global sea levels fell. This was a serious problem, because so much marine life lived in shallow water over the continental shelves. When the sea retreated, those habitats did not simply become slightly less comfortable. Many of them disappeared. Imagine an entire neighbourhood slowly losing its air, food supply, and available space at the same time. Moving would seem like the obvious solution, but marine organisms cannot always relocate quickly, especially when suitable habitats are shrinking across the planet. Species adapted to warm tropical water faced cooling temperatures. Animals living on shallow seafloors found those seafloors exposed above the water. Food webs were disrupted as conditions changed around them. The extinction appears to have happened in more than one pulse. The first was associated with cooling, glaciation, and falling sea levels. Later, as the climate warmed and the ice melted, sea levels rose again, but that did not simply return everything to normal. Changes in ocean circulation and oxygen levels placed new pressure on surviving species. Life had adjusted to one emergency and was then handed another before it had properly recovered. Earth, on this occasion, behaved like someone changing the rules halfway through a board game and then acting surprised when most of the players left. Evidence from ancient ocean temperatures indicates that a relatively short cooling pulse occurred near the end of the Ordovician and coincided with the glacial maximum and the extinction. The episode removed a large portion of marine species, making it the earliest of the 5 major extinction events recognised in the fossil record. It is tempting to picture this as a silent ocean becoming empty, but extinction does not work so neatly. Many species survived. Ecosystems continued. Some groups declined sharply, while others passed through with fewer losses. The seas did not become lifeless. They became less diverse, with many long-established communities broken apart. Recovery took millions of years. New species eventually appeared, and marine ecosystems were rebuilt through evolution. That does not mean the previous world returned. Evolution does not keep a backup copy. Once a species is gone, later organisms may occupy a similar role, but they are not the same species, and the relationships between them are different. Roughly 70 million years later, another major crisis developed during the late Devonian. This one was less like a single blow and more like a series of failures that unfolded over a long interval. The Devonian is sometimes called the Age of Fishes because fish became increasingly varied and important in the oceans. Armoured fish swam through marine habitats, early sharks were spreading, and lobe-finned fish included relatives of the animals that would eventually move onto land. The seas also contained enormous reef systems. These reefs were not built mainly by the same corals that dominate modern reefs. Much of the structure came from ancient corals and sponge-like organisms called stromatoporoids. Together, they created complex habitats that supported many other species. On land, something equally important was happening. Plants were becoming larger and more widespread. Early forests began to develop, complete with deeper roots and more complicated soils. This transformation of the continents was one of the great biological changes in Earth's history, but it may also have affected the oceans in ways that caused trouble. Roots broke down rock and changed the rate at which nutrients moved from land into rivers and seas. Increased nutrients may have encouraged large blooms of algae and other microorganisms. When those organisms died and decomposed, the process consumed oxygen in the water. In some places, deeper parts of the ocean became oxygen poor or completely anoxic. For animals that need dissolved oxygen, an anoxic ocean is not merely inconvenient, it is uninhabitable. Fish can sometimes swim away if oxygen levels fall gradually, and if better water is available nearby. Animals attached to reefs do not have that option. A brachiopod cannot gather its belongings and head for a more promising coastline. Reef systems are especially vulnerable because the organisms building them often require a narrow range of temperature, water chemistry, light, and oxygen conditions. The late Devonian crisis was probably driven by several interacting pressures rather than one universally accepted cause. Ocean anoxia played a major role. Climate changes, including periods of cooling, were also involved. Volcanic activity may have contributed, and changes caused by the spread of land plants could have altered nutrient cycles and atmospheric carbon dioxide. Scientists continue to study which processes mattered most and how they influenced one another. That uncertainty is not a sign that nothing is known. It reflects the difficulty of reconstructing a complex event that took place more than 370 million years ago. Rocks preserve chemical signals, fossils, and sediment patterns, but they do not provide a narrated explanation. Geologists must compare records from many locations and work out whether changes happened at the same time. It is rather like investigating a crime using several damaged calendars, a box of seashells, and some mud collected before anyone knew there would be questions. The late Devonian extinction affected marine ecosystems much more heavily than life on land. Reef-building organisms suffered severe losses, and the great reef systems that had flourished earlier in the period largely disappeared. Some groups of armoured fish declined dramatically. Many species of trilobites, brachiopods, and other marine animals vanished. Unlike the later asteroid extinction, the Devonian crisis does not fit comfortably into one dramatic day. Studies of the rock record show that major environmental disturbances and extinction pulses were separated in time, with important events occurring hundreds of thousands of years apart. The precise chain of causes remains debated, but the pattern shows a planet experiencing repeated biological stress rather than one isolated accident. The next crisis was the largest known mass extinction in the history of complex life. It occurred about 252 million years ago, at the end of the Permian period. Paleontologists sometimes call it the Great Dying, which is unusually direct language for scientists. They could have called it the Permian Biodiversity Reorganization Event, but even geology occasionally admits when things went very badly. Before the extinction, most of Earth's land had joined into the supercontinent Pangaea. Its enormous interior was often dry and highly seasonal. On land lived early relatives of mammals, along with amphibians, reptiles, insects, and a wide range of plants. The ocean supported reefs, shellfish, fish, ammonoids, and the remaining trilobites, a group that had survived for hundreds of millions of years. Then vast volcanic eruptions began in what is now Siberia. These eruptions created the Siberian Traps, an enormous region of volcanic rock. Lava spread across a huge area during repeated episodes. The lava itself was destructive locally, but the greater global danger came from gases released by the volcanic activity and by magma heating surrounding rocks. Large quantities of carbon dioxide entered the atmosphere. Global temperatures rose sharply. The warming changed rainfall, weathering, and ocean circulation. Carbon dioxide dissolved into seawater, affecting ocean chemistry and making it harder for some organisms to build shells and skeletons. Warm water holds less oxygen than cold water. At the same time, changing circulation can reduce the supply of oxygen to deeper parts of the sea. The oceans became increasingly anoxic. In some environments, microbes that thrive without oxygen may have produced hydrogen sulfide, a gas toxic to many forms of life. Researchers have found sulfur isotope evidence that oxygen-poor water repeatedly expanded into shallower marine environments during the crisis. Conditions on land were also harsh. Extreme heat, drought, acid rain, wildfires, and damaged vegetation placed ecosystems under sustained pressure. Soil erosion increased where plant cover was lost. Rivers carried more sediment and nutrients into the oceans, adding further stress to marine systems. The disaster was not caused by one simple mechanism. It was a cascade. Volcanism changed the atmosphere. The altered atmosphere warmed the climate. Warming affected the oceans. Ocean chemistry and oxygen levels shifted. Food webs weakened. Organisms already near their physiological limits became unable to cope. Tropical ocean temperatures may have become especially dangerous. An animal can adapt to gradual change over generations if enough individuals survive, and if the change stays within the range its biology can tolerate. There is no useful adaptation to water that becomes too hot to breathe in. At some point, enzymes stop functioning properly, oxygen demand rises while oxygen availability falls, and survival becomes impossible. Most marine species disappeared during the end-Permian crisis. On land, many vertebrate groups and plant communities were severely reduced. Trilobites, after surviving the first two major extinctions and several smaller crises, finally vanished completely. They had lasted for more than 250 million years. That is an impressive run, although it ended without a farewell tour. The immediate aftermath was not a quick return to healthy ecosystems. Environmental instability continued, and recovery was slow. For millions of years, marine and terrestrial communities remained relatively simple. Some hardy organisms became extremely common because they could tolerate the damaged conditions. These are sometimes called disaster taxa. A disaster taxon is not necessarily superior under normal circumstances. It is simply well suited to a disturbed world. In a stable ecosystem, specialists can thrive by using particular foods or habitats very efficiently. During a crisis, flexibility often matters more. An organism that can live in many environments and eat several kinds of food has a better chance than one requiring one exact type of reef, one narrow temperature range, and meals served promptly at 6. Life eventually recovered, but the Permian world did not return. New groups expanded into the empty ecological space. The Triassic period developed its own communities, including early dinosaurs, new marine reptiles, and the ancestors of later mammals. The extinction had removed many of the groups that once dominated the planet, allowing different ones to take their place. About 51 million years after the end-Permian event, another major extinction struck at the boundary between the Triassic and Jurassic periods. By then, dinosaurs had already evolved, but they were not yet the unquestioned rulers of the land. They shared ecosystems with crocodile relatives, large amphibians, mammal ancestors, and many other reptiles. Once again, immense volcanic activity was involved. Pangaea was beginning to split apart, and magma rose through the crust along the developing rift. The eruptions formed the Central Atlantic Magmatic Province, an area of igneous rock that can now be found across parts of North America, South America, Europe, and Africa. The eruptions occurred in pulses. Carbon dioxide and other gases entered the atmosphere, driving rapid climate changes. Volcanic sulfur could produce short periods of cooling, while carbon dioxide caused longer warming. The oceans absorbed carbon dioxide and became more acidic. Oxygen levels in marine environments changed, and repeated environmental shocks placed organisms under strain. Evidence from volcanic rocks and sediment layers shows a close timing between intrusive volcanic activity and the start of the end-Triassic extinction. Mercury spikes found in widely separated rocks also indicate repeated volcanic emissions during the biological crisis. Many large amphibians and non-dinosaurian reptiles disappeared. In the oceans, ammonoids, reef organisms, bivalves, and other groups suffered severe losses. Dinosaurs were affected too, but several dinosaur lineages survived. Once many competitors were removed, the surviving dinosaurs expanded into newly available habitats. This is an important pattern in mass extinctions. The survivors are not always the organisms that seemed most dominant before the crisis. Extinction changes the rules. Traits that were useful in the old environment may become liabilities. Traits that once seemed unremarkable can suddenly make the difference between survival and disappearance. After the end-Triassic event, dinosaurs diversified and became the dominant large land animals for most of the Jurassic and Cretaceous periods. Their success lasted about 135 million years. They spread across every continent and developed into an enormous variety of forms. Some were giant long-necked herbivores, some were armoured, some had horns, crests, clubs, or rows of plates. Small feathered dinosaurs gave rise to birds, which are still with us today, and remain perfectly willing to steal food from an unattended table. The fifth major extinction occurred 66 million years ago, at the end of the Cretaceous period. This is the one almost everyone knows, largely because it involved dinosaurs and an asteroid. If the same event had only affected several obscure families of marine worms, it might not have received quite as many films. A large asteroid struck Earth near the present-day Yucatan Peninsula in Mexico. It created the Chicxulub crater, which is roughly 180 to 200 kilometers wide. The impact released an immense amount of energy, causing earthquakes, tsunamis, and intense heat near the impact zone. Material blasted from the crater entered the atmosphere. Dust, sulfur-rich aerosols, and soot spread around the planet. Sunlight reaching the surface fell sharply. Temperatures dropped, and photosynthesis slowed or stopped in many environments. Plants and microscopic ocean plankton struggled, affecting the herbivores that ate them and the predators that ate those herbivores. The original impact hypothesis grew from the discovery of unusually high iridium concentrations in a thin layer of rock marking the boundary between the Cretaceous and Paleogene periods. Iridium is rare in Earth's crust, but more common in many asteroids. The researchers proposed that a major impact had spread the material worldwide and caused the extinction. Later discoveries, including shocked minerals, glassy droplets, and the Chicxulub crater itself, strengthened the case. The location of the impact made the consequences worse. The asteroid struck rocks containing sulfur and organic material. Vaporized sulfur contributed to aerosols that blocked sunlight, while soot added to the darkness and cooling. Research on material from the crater indicates that organic matter at the impact site helped intensify the impact winter. Large animals requiring a great deal of food were particularly vulnerable once food webs collapsed. The non-avian dinosaurs disappeared. Pterosaurs vanished. Many marine reptiles died out. Ammonites, once widespread and diverse, were lost. Numerous plankton species and other marine organisms also disappeared. Not every dinosaur went extinct. Birds are living dinosaurs, descended from small feathered theropods. Their survival may have been helped by small body size, flexible diets, and the ability of some species to eat seeds. Seeds can remain available after green vegetation has died back, making them a useful emergency food when fresh leaves and fruit are in short supply. Small mammals also survived. Many could shelter in burrows, reproduce relatively quickly, and eat varied diets. Some may have fed on insects, seeds, roots, or decaying material. Again, survival didn't necessarily favour the largest brain, strongest jaws, or most impressive appearance. Sometimes the best preparation for global catastrophe is being able to hide underground and eat whatever is available. Crocodilians, turtles, amphibians, and certain freshwater animals survived at higher rates than many land and ocean groups. Freshwater ecosystems may have received food from dead organic matter washed into rivers and lakes. Animals with slower metabolisms could survive longer without regular meals. A crocodile can wait patiently for food. A giant active predator has a more demanding schedule. Once the skies cleared and photosynthesis recovered, ecosystems began rebuilding. Ferns spread rapidly across damaged landscapes. Mammals diversified. Birds expanded into new forms. Over millions of years, forests returned and food webs became complex again. All 5 events were different, but they share several features. Each involved major environmental change occurring quickly enough to overwhelm the ability of many species to adapt or move. Climate shifts played a role in all of them, although the direction and causes differed. Ocean oxygen and chemistry were repeatedly important. Habitat loss spread through food webs. Multiple pressures often acted together. Only one of the 5 was primarily caused by a large asteroid impact. Volcanism is associated with several others. This does not mean volcanoes are generally bad for life. Ordinary volcanic activity creates land, releases nutrients, and contributes to long-term geological cycles. The danger comes when eruptions occur on an enormous scale and release gases faster than Earth's systems can absorb them. The same principle applies to climate change. Earth's climate has always changed, but speed matters. A slow shift gives species time to migrate, evolve, and adjust their relationships with other organisms. A rapid shift can separate plants from their pollinators, predators from prey, and animals from the climates they can tolerate. A few degrees of change over millions of years is very different from similar change over centuries or thousands of years. Scale matters too. A local environmental disaster can be devastating without becoming a mass extinction. A forest can burn, a lake can dry out, or a reef can collapse while the same species survive elsewhere. A global crisis removes those safe alternatives. When similar pressures affect many regions at once, there may be nowhere suitable to move. The fossil record also shows that ecosystems can begin failing before many species officially disappear. Abundant organisms become rare. Populations break into isolated groups. Reproduction falls. Food webs lose connections. By the time the last member of a species dies, its ecological role may have been absent for generations. This makes the past especially relevant to the question we began with. The Big Five were not simply moments when a large number of names disappeared from a list. They were periods when environmental conditions changed, common species became scarce, ecological relationships broke down, and extinction outpaced the creation of new species. The differences are just as important as the similarities. No previous mass extinction was caused by one species deliberately transforming land, moving organisms between continents, harvesting wildlife across every ocean, and changing the atmosphere through industrial activity. The present situation has no exact historical copy. That does not prevent comparison. The rock record tells us which pressures have caused large biological losses before. Rapid warming, rapid cooling, ocean acidification, oxygen loss, habitat destruction, disrupted nutrient cycles, and food web collapse all appear in different combinations. Several of those processes can already be measured today. The question is no longer limited to whether Earth is capable of another mass extinction. The planet has demonstrated that ability 5 times with considerable thoroughness. What we need to examine now is whether current changes are beginning to resemble the early stages of those older crises, how quickly modern extinction rates are rising, and whether calling the present period a 6th mass extinction is scientifically accurate, or still a warning about where we may be heading. That leaves us with a question that sounds as though it should have a simple yes or no answer. Are we already living through the 6th mass extinction? The most accurate response is that it depends on what we mean by living through it. If we mean that Earth has already lost at least 75% of its species, then no. The known modern losses have not reached the scale used to identify the 5 largest extinction events in the fossil record. If we mean that extinction rates have risen far above their normal level and could eventually produce a loss of that size, then many researchers argue that the process has already begun. Both answers can be correct because they measure different things. One looks at how much has already been lost. The other looks at how quickly loss is happening. Suppose a train has left its station and is heading toward another city. One person might say the journey is already underway because the train is moving. Another might say the destination has not been reached because the train is still several hours away. They are not necessarily disagreeing about the location of the train. They're using different points in the journey to define what counts as arrival. The same problem appears in discussions of the 6th mass extinction. Paleontologists often identify mass extinctions after examining the completed pattern in layers of rock. They can see how many species disappeared across an interval and compare the world before the event with the world after it. Modern biologists are standing inside the interval. They can measure population declines, habitat loss, rising threats, and documented extinctions, but they cannot examine the finished result because it has not happened yet. This makes the word already particularly troublesome. A future geologist might look back at our time and mark the beginning of a major extinction event somewhere around the expansion of agriculture, the spread of humans across continents and islands, the Industrial Revolution, or the period of rapid global change that began in the 20th century. From that future viewpoint, the answer might be obvious. From inside the process, the boundaries are much less tidy. The strict benchmark is useful. A mass extinction is generally described as the loss of at least 3/4 of species over a geologically short period. By that measure, the present crisis has not yet reached the same total magnitude as the Big Five. A major scientific review comparing modern losses with the fossil record concluded that recent extinction rates are unusually high, but that the 75% threshold had not yet been crossed. The same review warned that continued losses, especially among species already classified as threatened, could push the world toward that threshold. That might sound reassuring for a moment. 3/4 of species are not gone. Most ecosystems still contain life. Birds continue to visit gardens, fish remain in the oceans, and insects continue to enter houses despite receiving no formal invitation. The difficulty is that waiting for 75% of species to disappear before admitting that a mass extinction is underway would be a little like waiting for most of a forest to burn before deciding that the first flames were part of a wildfire. The final threshold tells us the scale of the completed event. It does not tell us that the earlier losses were harmless. To understand the present situation, scientists examine extinction rates. This means comparing the number of species disappearing now with the background rate expected during periods without a mass extinction. Background extinction is difficult to calculate precisely. The fossil record contains gaps. Species are defined differently in different studies. Marine animals with shells are more likely to become fossils than small, soft-bodied organisms. A species may have existed for thousands of years without leaving a trace that scientists have found. Estimating a normal extinction rate is therefore less like reading a complete register and more like reconstructing attendance from a badly damaged guest list. Researchers try to deal with these uncertainties by using conservative assumptions. One widely discussed study examined modern vertebrate extinctions using a background rate that was deliberately higher than many earlier estimates. Even with that cautious comparison, the researchers found that the average rate of vertebrate species lost during the previous century was as much as 100 times the background rate. They calculated that losses occurring over roughly a century would normally have taken between about 800 and 10,000 years, depending on the group being examined. This does not mean every group of organisms is disappearing at exactly 100 times its natural rate. Extinction rates vary between groups, locations, and methods of calculation. It does mean that the observed pace of loss is far outside what would usually be expected. Rate is why some researchers say the sixth mass extinction is already underway. They are not claiming that the final total has been reached. They are saying the process producing the total can be detected. There is another complication. Confirmed extinction is a very late signal. Before a species becomes globally extinct, it usually disappears from many local areas. Its population becomes smaller. Separate populations become isolated. Genetic diversity declines. Reproduction becomes less reliable. The species may stop performing its former ecological role even while a few individuals survive. Consider a pollinating insect that once lived across an entire region. It may still exist in one protected valley, so it is not extinct. Yet farms, forests, and wild plants outside that valley no longer receive its visits. From the point of view of the species list, it remains present. From the point of view of most ecosystems where it once worked, it is already absent. Global extinction is the final entry in a much longer record of decline. It is also the hardest stage to reverse, mostly because bringing back a species after the last individual has died presents several practical difficulties. Population loss can therefore provide an earlier and more sensitive warning than species extinction alone. A 2020 study examined nearly 29,400 terrestrial vertebrate species and identified 515 with fewer than 1,000 individuals remaining. For a subset of mammals and birds on the brink, the researchers estimated that about 94% of their populations had disappeared during the previous century. They argued that these population losses showed an accelerating crisis even before all of the affected species became extinct. A species with 900 individuals is still a species. It may even look reasonably secure if all 900 animals are gathered in one photograph, although convincing them to cooperate would be difficult. In biological terms, however, the situation can be dangerous. A small population is more vulnerable to disease, fire, storms, breeding problems, loss of genetic diversity, and ordinary bad luck. If those individuals are divided among several tiny populations, the risk increases. One group may contain too few breeding adults, another may have an uneven balance of males and females, a third may live in a habitat that is slowly disappearing. The total number alone cannot describe all of this. The International Union for Conservation of Nature tries to assess such risks through the Red List of threatened species. The Red List does not simply count how many animals remain. Its assessments consider population trends, geographic range, fragmentation, observed decline, projected threats, and other evidence. Species may be classified as vulnerable, endangered, or critically endangered depending on the level of risk. The 2026-1 Red List reports more than 48,600 species as threatened with extinction, representing about 28% of assessed species. Among groups with relatively comprehensive assessments, the best estimates indicate that roughly 71% of cycads, 44% of reef-forming corals, 41% of amphibians, 38% of trees, 38% of sharks, rays and chimeras, 26% of mammals, 26% of freshwater fishes, 21% of reptiles, and around 11.5% of birds are threatened. These percentages should not be combined into one simple global average because the level of assessment differs greatly among groups. That final limitation matters. Extinction risk has been evaluated for less than 5% of the world's described species. Mammals and birds are relatively well studied. Many insects, fungi, marine invertebrates, and other organisms are not. The animals most familiar to us are not necessarily the ones that best represent the full condition of life on Earth. This creates an unusual situation. We know enough to see serious decline, but not enough to count every loss. There could be threatened species that have not been assessed, declining populations that have not been monitored, and extinctions that have not been recorded. A small insect living in one patch of forest can disappear without a research team ever learning that it existed. It receives no final survey, no official announcement, and certainly no documentary narrated by someone whispering thoughtfully beside a tree. At the same time, threatened does not mean doomed. A threatened species has an elevated risk of extinction. It is not guaranteed to disappear. Categories can improve when habitats are protected, hunting is controlled, pollution is reduced, invasive species are removed, or breeding programs succeed. They can also worsen when threats increase. This distinction prevents two opposite mistakes. The first is assuming that tens of thousands of threatened species are already extinct. They are not. The second is assuming that because they still exist, the crisis is exaggerated. A patient in intensive care is alive, but this does not make intensive care an unnecessary hobby. Some groups provide especially clear evidence of a broad decline. Amphibians are one example. Frogs, salamanders, newts, and caecilians are affected by habitat destruction, disease, pollution, exploitation, and changing climate conditions. Many have small ranges and depend on moisture, temperature, and water quality remaining within narrow limits. The second global amphibian assessment evaluated more than 8,000 species and estimated that about 41% were globally threatened. It also found that the overall extinction risk for amphibians continued to worsen. Disease and habitat loss were major causes of earlier deterioration, while climate change became an increasingly important driver after 2004. Conservation produced genuine improvements for some species, showing that decline can be reversed, but those gains were smaller than the broader movement toward higher risk. Amphibians are not just unfortunate exceptions. They show how several pressures can combine. A frog may already occupy a limited mountain habitat. Agriculture removes part of the forest. A fungal disease reduces the remaining population. Warmer or drier conditions make reproduction less successful. A road divides the habitat and prevents movement between streams. No single pressure needs to kill every frog directly. Together, they can make recovery increasingly unlikely. That combination of pressures is common across modern biodiversity loss. The major direct drivers identified by the global IPBES assessment are changes in land and sea use, direct exploitation of organisms, climate change, pollution, and invasive alien species. Their importance varies by ecosystem, but they frequently overlap. The assessment concluded that extinction rates were already at least tens to hundreds of times higher than the average over the previous 10 million years, and estimated that around 1 million animal and plant species could face extinction, many within decades, unless the intensity of the drivers is reduced. The estimate of 1 million species does not mean scientists have identified 1 million species that will definitely vanish on a known schedule. It is an extrapolation from the best studied groups, habitat trends, extinction risk assessments, and the estimated total number of species. There is uncertainty in the number, but uncertainty does not turn the central finding into a guess. It means the total is presented as an estimate rather than an exact count. The modern crisis also differs from earlier mass extinctions in one major way. Its primary causes can be traced to the activity of a single species. Humans have transformed land for farming, settlement, mining, transport, and industry. We harvest animals and plants from land and sea. We move species between regions that were previously separated. We release pollutants. We change the composition of the atmosphere and climate. Each action may appear local when viewed alone, but together they reach nearly every ecosystem. This does not mean every person contributes equally. The environmental impact of a subsistence farmer is not the same as that of a large industrial operation. Consumption levels, technologies, laws, trade systems, wealth, and political power all shape the pressure placed on nature. Saying the crisis is human-caused should not be used to turn 8 billion people into one identical environmental character with one enormous carbon footprint and a suspiciously large shopping trolley. It means the causes arise from human systems rather than from an asteroid impact or a continent-sized volcanic province. Because those systems can change, the final outcome is not predetermined. That is one reason the word extinction can mislead. It sounds complete and irreversible, because extinction itself is both. The process leading toward extinction is not always irreversible. A population can grow, a forest can regenerate, a wetland can be restored, a species can return to an area from which it disappeared. Recovery may be difficult, slow, and expensive. Some damage cannot be repaired within a human lifetime. A mature forest cannot be recreated by planting rows of young trees and returning on Tuesday to see whether the owls have moved in. Even so, conservation can prevent losses that would otherwise occur. The current Red List itself contains species whose status has improved because of conservation. The amphibian assessment found that habitat protection and improved habitat management contributed to many genuine improvements. These cases do not cancel the declines, but they demonstrate that the direction can change when the causes are addressed. This leads to the central disagreement about naming the present period. Researchers who prefer the phrase sixth mass extinction often focus on the abnormal rate of loss, the number of populations disappearing, and the growing number of species pushed toward the edge. To them, waiting until the 75% threshold is reached would describe the crisis too late to prevent it. Researchers who use more cautious wording may accept the same evidence, while reserving mass extinction for the completed scale visible in the fossil record. They might call the present situation a biodiversity crisis, an extinction crisis, or the beginning of a possible mass extinction. Their caution is usually about comparison and definition, not a claim that species are doing well. Modern data and fossil data are also difficult to compare directly. Fossils usually allow scientists to identify losses across long intervals, often with limited detail. Modern monitoring can record changes from year to year, sometimes down to individual animals. If modern data were compressed into blocks lasting hundreds of thousands of years, the pattern might look different. If the fossil record could be observed with wildlife cameras, satellite images, genetic samples, and thousands of researchers carrying clipboards, our estimates of ancient extinction rates might also change. Species definitions create another difficulty. Paleontologists often work with morphological species identified from shells, teeth, bones, or other preserved structures. Modern biologists can use genetics, behavior, breeding patterns, anatomy, and ecology. Two populations that appear identical in the fossil record might be recognized as separate species today. Comparing the totals requires assumptions about what is being counted. These technical issues are important, but they do not remove the modern signal. Whether current extinction is calculated with generous or conservative background rates, the loss of vertebrate species is unusually rapid. Populations are declining across many groups. Habitat is being altered on a global scale. Extinction risk is increasing faster than successful recovery. So are we already living through the sixth mass extinction? Under the strictest definition based on total species lost, not yet. Under a definition based on the beginning of an exceptionally rapid, human-driven extinction process, there is a strong scientific case for saying yes. A useful middle answer is that we are living through a global extinction crisis with the potential to become the sixth mass extinction. That wording does not reduce the seriousness of the situation. It describes both what is known and what remains undecided. The rate is already dangerous. The final magnitude depends on how long the rate continues and whether the pressures become stronger or weaker. This is not a dispute between alarm and calm. Calm scientific language can describe a severe problem. Alarmed language can sometimes describe it poorly. The goal is not to choose the most frightening phrase, it is to understand what the measurements are telling us. One measurement says that the completed loss has not reached 75%. Another says extinction is occurring much faster than the long-term background rate. Another shows that tens of thousands of assessed species face elevated risk. Another shows that many populations and local ranges have already disappeared. Another shows that some conservation efforts work. Taken together, these findings describe a crisis that is underway but not finished. They also tell us that the future cannot be read from the current extinction count alone. A species may decline for decades before vanishing. An ecosystem may lose function before losing every member. Damage that seemed gradual can accelerate when populations become small and ecological connections begin to break. For that reason, the clearest signs are not always found in announcements that another species has officially become extinct. They appear earlier in quieter changes. Fewer insects emerging in spring, breeding seasons shifting, coral cover declining, migratory animals arriving at the wrong time, forests losing large seed-dispersing animals, rivers becoming warmer, and once common species surviving only in scattered pockets. Those are the signals that reveal whether a living system is merely changing, as ecosystems always have, or being pushed faster than its species can respond. To understand how close the world may be to a larger collapse, we need to examine those warning signs before they become final entries on an extinction list. Those warning signs rarely announce themselves with a clear beginning. There is no single morning when a forest becomes endangered, no official bell when an ocean food web starts to fail, and no notice placed beside a river explaining that its biodiversity has entered a difficult period. Most ecological decline appears as a collection of smaller changes. A population becomes less common. A breeding season produces fewer young. A species disappears from one valley but remains in the next. A reef survives a hot summer but has not recovered when another hot summer arrives. This is why scientists do not wait only for confirmed extinctions. Extinction is the final result, and by then the useful warning period has passed. To understand the condition of the living world, researchers track abundance, distribution, reproduction, migration, seasonal timing, habitat quality, genetic diversity, and the relationships between species. Each measurement provides a different piece of information. No single one proves that a mass extinction is approaching, but several worsening together can reveal that an ecosystem is losing its ability to cope. The most basic signal is a decline in abundance. This simply means there are fewer individuals of a species than there were before. A bird may still be found across the same country, but its population may have fallen from several million to a few 100,000. On a map, the species looks present. In the field, it is becoming harder to find. Abundance matters because common species often do much of the daily work in an ecosystem. They move seeds, pollinate flowers, consume vegetation, control prey populations, recycle nutrients, and provide food for other animals. A rare species may be scientifically important, but a common species can have a larger effect on the movement of energy through an ecosystem simply because there are so many individuals performing the same role. Long-term insect studies provide a useful example. A global analysis of hundreds of terrestrial insect communities found overall declines in abundance and weaker declines in species richness. The strongest average losses were often among species that had been abundant at the beginning of the monitoring period. The rare species attracted attention because they were already unusual, but the reduction of familiar numerous insects accounted for much of the overall decline. This can be difficult to notice without careful records. Human memory is not an ideal scientific instrument, especially when asked to recall the number of moths around a light 20 years ago. People may remember that there used to be more butterflies, but they may not remember the weather, season, location, or exact species. Standardized monitoring is needed to separate a real long-term trend from ordinary variation. Insect populations can change sharply from one year to another. A wet spring may help one group and harm another. A dry summer may reduce larvae that depend on ponds while benefiting insects that prefer warm grassland. A cold week during breeding season can produce a poor year without indicating a permanent decline. That is why researchers need repeated surveys using similar methods over long periods. The warning appears when low numbers stop being a temporary fluctuation and become the new normal. One disappointing breeding season is part of nature. 10 or 20 disappointing seasons, especially across many locations, suggest a deeper problem. The same principle applies to fish surveys, bird counts, plant plots, coral monitoring, and almost every other form of ecological observation. A second signal is range contraction. A species does not need to vanish globally to lose most of its former world. It may disappear first from the warmest part of its range, from heavily developed areas, or from isolated patches where populations are too small to recover after disturbance. The remaining individuals become concentrated in fewer places. Range contraction increases risk because the species is no longer spreading that risk across many populations. A storm, wildfire, disease outbreak, or new predator can affect a much larger share of the remaining population at once. It is the ecological equivalent of keeping every spare key in the same drawer. Everything seems manageable until the drawer disappears. Some species respond to warming by moving toward the poles, uphill, into deeper water, or into cooler local habitats. These shifts have been documented across terrestrial and marine environments, although the direction and speed vary widely. A broad review of recorded range changes found that many were consistent with changing climate, while others were slowed, redirected, or prevented by habitat conditions, species interactions, and barriers to movement. Movement can look like successful adaptation, and sometimes it is. A mobile animal may colonize new habitat and maintain a healthy population. A plant may spread its seeds into a cooler region. A fish may move farther north and continue reproducing. The full picture is usually more complicated. Expansion at the cool edge of a range can occur at the same time as decline at the warm edge. The species appears in new places, but the total population may still shrink. Research on North American birds found that many species shifted their ranges over 2 decades, yet those movements only partly reduced their exposure to warming. Even highly mobile birds could not always move quickly or far enough to remain within their previous climate conditions. Less mobile species face greater difficulties. A mountain plant cannot move uphill if it already lives near the summit. A freshwater fish cannot leave a warming river and cross dry land to another watershed. A forest animal may be capable of travelling many kilometres, but still be unable to cross highways, cities, farmland, or fences. Suitable climate is not enough. A species also needs suitable soil, food, shelter, nesting sites, breeding partners, and protection from predators or competitors. A butterfly following cooler temperatures may arrive in an area where its caterpillar's food plant does not grow. The butterfly has technically reached the right climate and still has nowhere useful to live. This is why fragmented habitats are an important warning sign. A large continuous forest can support movement between populations. Divide it into small patches, and the distance between those populations becomes harder to cross. Roads, farms, dams, walls, and urban development can turn one connected habitat into a set of biological islands. Isolation gradually changes populations even before their numbers become extremely low. Individuals have fewer opportunities to breed with animals or plants from other groups. Genetic diversity may decline, harmful genetic traits can become more common, and the population may have less variation available to help it adjust to disease, heat, drought, or other environmental changes. genetic decline is nearly invisible without laboratory work. A forest may still contain the same number of trees while becoming less genetically diverse. A small population of mammals may continue producing young while becoming increasingly related. The outward appearance can remain normal for years, rather like a family gathering where everyone looks healthy, but the guest list has become worryingly repetitive. Reproduction provides another early warning. Counting adults alone can hide a population that is no longer replacing itself. Long-lived animals and plants may remain present long after breeding conditions have deteriorated. A forest of mature trees can survive for decades, even if almost no seedlings are reaching adulthood. A turtle population may contain many old adults, but very few young turtles. Ecologists therefore study age structure. A healthy population usually contains individuals at several life stages. The exact balance varies by species, but a persistent absence of young animals or new plants suggests that the population may decline later, even if the adults appear stable now. The delay can create false reassurance. Large trees do not vanish immediately when pollinators decline. Adult seabirds may return to the same nesting cliffs for many years, despite producing fewer surviving chicks. Coral colonies may remain physically present after repeated stress has reduced growth and reproduction. What looks like stability may be an extinction debt waiting to be collected. Seasonal timing can reveal problems that a simple count misses. Living things use temperature, rainfall, daylight, snowmelt, and other signals to decide when to flower, migrate, breed, hatch, or emerge. These recurring events are known as phenology. Climate change can shift those schedules, but species do not all respond in the same way. A plant may flower earlier during a warm spring. Its pollinating insect may respond to a different temperature signal or to the length of the day. A migratory bird may begin its journey according to conditions thousands of kilometers away. By the time it reaches its breeding ground, the peak supply of caterpillars may already have passed. A large global analysis of terrestrial observations found increasing differences between the seasonal timing of plants and animals from 1981 to 2020. Plants and animals often change their schedules at different rates because they rely on different cues and have different biological limits. Extreme weather can make this even less predictable. Research across the United States found that unusual heat, drought, cold, and heavy rainfall affected the flowering times of plants and the flight periods of butterflies and moths. Plants and insects sometimes responded in different directions to the same event, increasing the chance that their periods of activity would no longer overlap properly. A mismatch does not automatically produce collapse. Some species can change their behaviour, switch food, extend their breeding period, or use several pollinators. Ecological relationships are often flexible. Nature has survived many early springs without submitting a formal complaint. The danger increases when a species depends heavily on a short seasonal event. Young birds may need large numbers of insects during a narrow growth period. A pollinator may depend on flowers that open for only a few weeks. Fish larvae may need plankton of the correct size soon after hatching. Missing that food peak can reduce survival for an entire generation. Repeated timing problems can weaken populations slowly. There may still be enough food for some individuals, so no dramatic die-off occurs. Instead, fewer young survive each year. Adult numbers decline later. By the time the pattern becomes obvious, the mismatch may have been affecting reproduction for decades. Coral reefs display a more visible warning. When seawater remains unusually warm, corals can expel the microscopic algae living inside their tissues. These algae provide much of the coral's energy and give it much of its colour. Without them, the coral appears white, a condition known as bleaching. Bleaching is not the same as immediate death. A coral can recover if temperatures return to safer levels and if other conditions remain favourable. The real danger comes from severe heat, long exposure, and repeated events that leave too little recovery time. Marine heat waves are becoming more frequent and intense, and they can change not only coral survival, but the wider structure of reef communities. Bleached and damaged corals affect microorganisms, fish habitat, food availability, and the physical structure that protects coastlines and supports many other species. Global field records show that recovery varies greatly among reefs, coral types, depths, and local conditions. Some reefs recover well after a heat wave, while others lose coral cover or change into a different ecological state. The Great Barrier Reef shows why repeated disturbance matters. Research reconstructing several centuries of regional ocean temperatures found that recent heat extremes were exceptional in that long record and were associated with repeated mass bleaching. The concern is not only that corals bleach once, it is that warming can make bleaching conditions occur so often that slower growing corals cannot rebuild before the next event. Recovery time is therefore a warning sign in its own right. Forests, reefs, wetlands and grasslands have always experienced disturbances. Fires burn, storms break trees, rivers flood, and predators cause prey populations to fall. Healthy ecosystems often recover because surviving organisms reproduce, neighbouring populations move in, and damaged habitats rebuild. Trouble begins when disturbances arrive faster than recovery. A forest that burns once may regenerate. The same forest may change permanently if it burns again before young trees mature. A reef may survive one marine heatwave but lose its main builders after several closely spaced events. A seabird colony may recover from one poor breeding year but not from a decade of reduced food. Scientists call this loss of resilience. Resilience does not mean an ecosystem remains unchanged. It means the system can absorb disturbance without losing its main structure and functions. When resilience falls, a relatively ordinary event can push the ecosystem into a different condition. A clear lake can become dominated by algae. A coral reef can shift toward algae-covered rubble. A forest can become open scrubland. A grassland can lose plant diversity after changes in grazing, fire, rainfall, or predator numbers. Returning the original conditions may then be difficult because the new system begins reinforcing itself. Freshwater environments contain many of the strongest warning signals. Rivers, lakes, streams, marshes, and ponds occupy a small part of Earth's surface, but they support a large amount of biodiversity. They are also directly affected by water extraction, dams, pollution, sediment, invasive species, overharvesting, and warming. A global assessment of freshwater fish, dragonflies, damselflies, crabs, crayfish, and shrimp concluded that roughly 1/4 of assessed freshwater species face a high risk of extinction. Pollution, dams, water extraction, agriculture, and invasive species were among the major threats. The study also emphasised that entire river catchments must be considered, because a protected patch downstream cannot remain healthy if the water arriving from upstream carries pollutants or has been diverted elsewhere. Freshwater decline may first appear through changes in the community rather than complete species loss. Sensitive insects disappear while pollution-tolerant species remain. Large fish decline while smaller, faster-breeding fish increase. Clearwater plants are replaced by algae. The river still contains life, but the surviving community indicates poorer water quality and simpler ecological conditions. Some recovery is possible when sewage treatment improves, barriers are removed, and habitats are restored. European freshwater monitoring recorded gains in invertebrate richness and abundance during earlier decades following water quality improvements. Those gains later slowed or stopped at many sites, particularly near dams, cities, cropland, and faster warming waters. The pattern shows both that restoration works and that older solutions may be insufficient when new pressures continue to increase. Another warning is biological homogenization. This happens when different places become more similar because distinctive local species decline while widespread, adaptable species expand. A woodland, wetland, and city park may each retain the same number of species, but that number can hide a major change. If unique specialists disappear and are replaced by the same common generalists, local species counts may remain stable, while regional diversity falls. Each location still looks reasonably lively, but the differences between them have been reduced. Long-term research on birds, butterflies and plants in Great Britain found that climate and land use change could increase local richness in some places, while also producing warmer adapted communities and greater similarity between locations. This is why counting species at one site does not always reveal whether biodiversity is improving. The identity of the species matters, as do their abundance and ecological roles. The replacement species are not bad simply because they are common. A pigeon is not personally responsible for global biodiversity decline, despite its occasionally smug expression. Generalist species succeed because they tolerate disturbed habitats, varied diets, and close contact with people. Their success becomes a warning when it occurs alongside the disappearance of specialists that cannot use the altered environment. Food webs provide another set of clues. The decline of one species can alter populations several steps away. Remove a predator, and its prey may become more abundant. Increased prey can reduce vegetation or smaller animals, remove an important herbivore, and fast-growing plants may crowd out others. Lose a seed-dispersing animal, and large-fruited trees may struggle to reproduce far from their parents. These effects are called trophic cascades, when changes move through several levels of a food web. They are difficult to predict because species rarely have only one relationship. A predator eats several prey species. A plant uses several pollinators. A fish may change diet as it grows. The network contains alternative routes, but those alternatives are not unlimited. Early food web failure may appear as unusual population swings. One species becomes extremely abundant while another collapses. Algal blooms increase. Herbivores damage vegetation that previously recovered. Predators travel farther for food or abandon breeding sites. The ecosystem has not become empty. Its controls are becoming less reliable. Scientists are especially concerned when several warning signs appear together. A population may survive habitat loss if its climate remains suitable. It may survive warming if it can move. It may survive disease if its population is large and genetically varied. Combine habitat fragmentation, warming, disease, and reduced reproduction, and each pressure makes the others harder to withstand. The IUCN Red List Index is designed to separate genuine changes in extinction risk from changes caused merely by assessing more species or improving scientific knowledge. Its trends show that several well-monitored groups have continued moving toward greater extinction risk, with reef-forming corals deteriorating particularly rapidly, and amphibians remaining among the most threatened animal groups. Not every warning sign is worsening everywhere. Some populations are growing. Certain species are expanding their ranges. Restored wetlands can recover birds, fish and plants. Protected habitats can support increasing numbers of large mammals. Cleaner rivers can regain insects that were absent for decades. These improvements are not exceptions that disprove the wider crisis. They help scientists identify which actions work. A species recovering after hunting stops tells us that exploitation was important. Fish returning after a dam is removed show that river connectivity matters. Birds increasing after pesticide restrictions demonstrate that chemical exposure can be reduced. The opposite lesson also applies. A protected species may continue declining if its habitat is warming, its prey is disappearing, or pollution arrives from outside the protected area. Conservation aimed at one pressure may fail when several others remain. This is why warning signs have to be read together. Fewer individuals, shrinking ranges, poor reproduction, changing seasonal timing, repeated bleaching, fragmented habitats, declining genetic diversity, slower recovery, simplified food webs, and increasing similarity between ecosystems all describe different parts of the same process. None of them provides a universal countdown. A population can decline and later recover. A range shift can create new opportunities. A bleached reef can regain living coral. Ecological change is not automatically extinction. The concern grows when the changes are widespread. persistent and driven by pressures that continue to intensify. At that point, the question is no longer whether one particular bird, insect, fish, or tree can survive one difficult season. It becomes whether enough species can keep adapting while the land, climate, oceans, and movement of organisms are all changing around them. Reading the warning signs tells us where the damage is appearing. It does not fully explain why it is happening. For that, we have to look more closely at the forces acting on species at the same time. The removal of habitat, the direct harvesting of wildlife, the arrival of invasive organisms, the spread of pollution and disease, and the climate changes that increasingly affect every other pressure. Those forces do not act separately. A forest may be cleared while the remaining trees become hotter and drier. A river may be polluted while a dam blocks migration. A seabird may lose its nesting habitat while the fish it eats are being harvested farther offshore. A frog may face a fungal disease in a landscape where wetlands have already been drained and rainfall has become less reliable. When several pressures arrive together, each one reduces the ability of a species to cope with the others. This is one of the main reasons modern biodiversity loss can move faster than it first appears. A species that might survive one disturbance may not survive 4 disturbances occurring at the same time. The largest direct pressure on many land and freshwater species is habitat change. Every organism needs a place where it can find food, reproduce, avoid danger, and complete the ordinary tasks required to remain alive. For a human, habitat can sound like scenery. For a wild species, it is the full set of physical conditions and relationships that make survival possible. A forest is not interchangeable with any collection of trees. An old forest may contain hollow trunks used by nesting birds, deep leaf litter where insects develop, fungi connected to roots, shaded streams, fallen wood, and plants adapted to several layers of light. Cutting it down and planting a row of young trees does not immediately rebuild those features. The new trees may absorb carbon and provide some shelter, but a plantation made from 1 or 2 species is not the same system as the forest it replaced. A woodpecker cannot nest in a sapling simply because someone has placed it on a tree planting spreadsheet. Habitat loss occurs for many reasons. Forests are cleared for agriculture, grazing, roads, mines, timber, housing, and industry. Wetlands are drained. Rivers are straightened or dammed. Grasslands are converted into crop fields. Coastal areas are developed for ports, resorts, and expanding cities. Seafloors can be damaged by fishing equipment, dredging, extraction, and construction. Agriculture is especially important because food production requires large areas of land and water. This does not mean farming is unnecessary. Humans need food, which remains one of our more persistent habits. The problem is how much land is used, which habitats are converted, what is grown, how water is managed, and whether wildlife can continue living around or within agricultural landscapes. A farm can contain hedgerows, ponds, trees, uncultivated margins, varied crops, and nesting areas. It can also be a very large field treated as though any insect entering without permission is conducting a hostile operation. The ecological difference between those systems can be substantial. Some species adapt to farmland. Others depend on conditions that farming removes. A bird that nests in open fields may benefit from certain forms of agriculture, while a forest specialist loses nearly everything it requires. Even species that initially survive can decline if breeding sites disappear, pesticides reduce food, or harvesting occurs during nesting seasons. Habitat loss is usually discussed in terms of area, but quality matters as well. A forest can remain visible on a satellite image while hunting, logging, fire, pollution, and invasive species steadily remove its ecological value. This is sometimes described as habitat degradation. A degraded habitat still exists, but it supports fewer species or provides poorer conditions. A wetland receiving fertilizer runoff may remain wet while becoming dominated by algae. A coral reef may retain its shape after many corals have died. a grassland may still look green while containing fewer native plants and insects. Fragmentation adds another problem. When a large habitat is divided into smaller pieces, each remaining patch develops more edges. The conditions near an edge are often different from those in the interior. Light, wind, temperature, humidity, predators, invasive species, and human disturbance can all change. A small forest surrounded by roads and fields may therefore contain much less true interior habitat than its total area suggests. Species that avoid edges are pushed into an even smaller central space. Those willing to cross open ground may still be killed by vehicles, exposed to predators, or unable to find the next patch. Roads are particularly effective at dividing habitat because they do more than occupy a narrow strip of land. They bring traffic, noise, artificial light, pollution, and access for further development. They can also alter drainage and create barriers for small animals. To a person driving along a road, the forest may appear continuous on both sides. To a salamander trying to reach a breeding pond, the road may be a wide, dry surface filled with fast-moving machinery and no obvious pedestrian crossing. In rivers, dams create a similar barrier. Migratory fish may be unable to reach spawning grounds, sediment becomes trapped upstream, water temperature and flow patterns change. Floodplains no longer flood naturally. Species below the dam receive a river that operates according to electricity demand or irrigation schedules rather than seasonal rainfall. Some dams provide important benefits to people, including power, water storage, and flood control. The ecological issue is not that every dam is identical or unnecessary. It is that changing the movement of water changes the system built around that movement. The next major pressure is direct exploitation. This means taking wild organisms faster than their populations can replace themselves. It includes hunting, fishing, logging, plant collection, and the wildlife trade. Humans have harvested wild species for thousands of years. Many forms of hunting, fishing, and gathering can continue without causing extinction when the number removed remains within the population's capacity to recover. The danger comes when demand, technology, and access increase faster than reproduction. A large animal may take years to reach maturity and produce only a few young. Removing breeding adults can cause a decline even when some individuals remain. The population may appear substantial until the missing generations begin to matter. Industrial fishing shows how technology can change the scale of exploitation. Modern vessels can travel farther, remain at sea longer, locate fish electronically, and process catches on board. A fish population that once had natural protection through distance, depth, or uncertainty can become much easier to find. Fishing does not affect only the species being targeted. Nets, lines, and traps may catch seabirds, turtles, sharks, marine mammals, and other animals by accident. Seafloor trawling can damage habitats used by many species. Removing large predatory fish can change the balance of entire food webs. Bycatch is a particularly unfortunate term because it makes the unintended animals sound like extra items placed in a shopping bag. For the animal involved, the distinction between target catch and bycatch is not especially meaningful. Hunting on land can also create ecological effects before a species disappears. Large animals are often important seed dispersers, grazers, browsers, predators, or scavengers. A forest may retain its trees after large mammals are removed, but the way seeds move and plants regenerate can change. This has led to the phrase empty forest syndrome. The trees remain, so the habitat looks intact from a distance, but many large animals have disappeared. The forest is physically present while losing part of its ecological machinery. Wildlife trade increases pressure when rare species become valuable, precisely because they are rare. This creates a dangerous feedback. As an animal becomes harder to find, its market value may rise, encouraging more effort to capture the remaining individuals. The same problem affects certain plants, reptiles, birds, fish, and mammals collected for pets, medicine, food, decoration, or status. Legal trade can be difficult to separate from illegal trade when documentation is poor or wild animals are presented as captive bred. Demand can sometimes fall quickly when laws, enforcement, public attitudes, and alternatives change. It can also move from one species to another. Protecting a heavily exploited species without addressing the reason people want it may simply redirect pressure toward a substitute. Invasive species form another major driver. An invasive species is not merely an organism living outside its original range. Many introduced species remain limited or cause little harm. The term is generally used for non-native organisms that spread and damage ecosystems, economies, or human health. Species have always moved, but humans now transport them across barriers they could rarely cross on their own. Ships carry organisms in ballast water and on hulls. Plants and animals are traded. Insects travel in timber and packaging. Seeds cling to equipment. Pets escape or are released when their owners discover that a small interesting animal has become a large interesting problem. On islands, introduced predators can be devastating. Many island birds evolved without land mammals that hunted eggs, chicks, or adults. They may nest on the ground, show little fear, and reproduce slowly. The arrival of rats, cats, snakes, or other predators can reduce their populations very quickly. The introduced predator is not behaving unnaturally. A rat finding an unguarded nest is simply having an unexpectedly successful afternoon. The problem is that the prey species did not evolve the defenses needed for that interaction. Invasive plants can change fire patterns, water use, soil chemistry, and the structure of habitats. Invasive insects and fungi can kill dominant tree species. Aquatic invaders can block waterways, filter large amounts of plankton, alter food webs, or compete with native species. Some invasions are easy to see. Others involve pathogens, parasites, or microorganisms that spread without attracting attention until native populations begin collapsing. The chytrid fungi affecting amphibians are a severe example. International movement of animals helped spread disease into populations with little resistance. Disease is a normal part of ecosystems, and pathogens do not automatically cause extinction. Healthy, connected populations often survive outbreaks because some individuals resist infection and recolonize affected areas. Disease becomes more dangerous when populations are already small, fragmented, stressed, or exposed to new pathogens. Climate conditions can also influence disease. Temperature and moisture affect the pathogen, the host, and their interactions. Warming may allow some diseases or their carriers to reach new regions. In other cases, heat may reduce transmission. There is no single rule that every disease becomes worse in a warmer world, but changing conditions can rearrange where and when infections occur. Pollution adds another wide group of pressures. The word covers many different substances and forms of disturbance, including pesticides, fertilizers, plastics, heavy metals, industrial chemicals, oil, sewage, noise, heat, and artificial light. Nutrient pollution often begins with nitrogen and phosphorus from fertilizers or waste entering rivers, lakes, and coastal waters. These nutrients encourage rapid growth of algae. When the algae die, decomposition uses oxygen. Fish and other animals may suffocate as oxygen levels fall. The water can look extremely productive because it is full of living algae, while actually becoming less capable of supporting its previous community. More life in one form can mean less life overall. Pesticides are designed to kill organisms considered harmful to crops, homes, or health. Their effects depend on the chemical, dose, timing, method of use, and which organisms are exposed. Some break down quickly, others persist. Some affect a narrow group, while others harm a wider range of insects, plants, birds, or aquatic animals. Even when a pesticide does not kill a bird directly, it may reduce the insects the bird feeds to its chicks. It can also affect navigation, reproduction, growth, or behavior at doses below immediate lethality. Ecological harm is not limited to animals dropping dramatically from the sky, which is fortunate because science would be much easier if every problem announced itself that clearly. Plastic pollution operates at several scales. Large debris can entangle animals or be swallowed. Smaller fragments enter food webs and spread through water, soil, air and living tissue. Chemicals associated with plastics can also cause concern. The full ecological effects of microplastics vary by concentration, organism, particle type and environment, and scientists are still working out which impacts are most important in natural conditions. The uncertainty is not about whether plastic is widespread, it is about how that exposure translates into population-level damage across different species. Noise pollution affects animals that depend on sound. Whales, dolphins, birds, frogs, insects, and many other species use acoustic signals to communicate, find mates, defend territory, locate prey, or avoid predators. Traffic, shipping, construction, aircraft, and industrial activity can mask those signals. An animal may call more loudly, change frequency, move away, or communicate at a different time. Adaptation can help, but it may also require extra energy or reduce the distance over which signals are heard. Artificial light changes the night environment. It can attract migrating birds, insects, and young sea turtles. It can alter feeding, reproduction, predator-prey relationships, and daily rhythms. Light does not need to kill an animal directly to reduce its chances of surviving or breeding. For a moth circling a lamp, the problem may look small. Across millions of lights and many generations, small changes in behavior can become an ecological pressure. Climate change increasingly interacts with all of these drivers. Rising average temperatures matter, but climate change is not simply a gradual warming of every place by the same amount. It changes extremes, rainfall, drought, snow, storms, fire conditions, ocean heat, sea level, and seasonal timing. Species are adapted not only to average conditions but also to limits. A plant may survive an average summer that is slightly warmer while dying during one short heat wave. A fish may tolerate gradual warming until water temperature crosses the point at which its metabolism requires more oxygen than the water can provide. Extreme events can therefore cause sudden losses inside a slower trend. Marine heat waves bleach coral. Drought weakens trees and makes them vulnerable to insects and fire. Heat can cause mass mortality in bats, birds, fish, and other animals. Floods may destroy nests or breeding sites. One extreme event does not necessarily create permanent decline. Populations often recover. The concern is that repeated events leave less time for recovery and may strike populations already reduced by other pressures. Sea level rise removes coastal habitat, especially where development prevents it from shifting inland. Saltwater enters freshwater wetlands. Nesting beaches become narrower. Low islands face erosion and flooding. A natural coastline can sometimes move as sea level changes. A wetland may gradually develop farther inland. A seawall, road, hotel, or city can block that movement. The habitat is squeezed between rising water and fixed development, a process sometimes called coastal squeeze. It is an accurate term and also sounds like an exercise no coastline volunteered to perform. Ocean warming affects where marine species live and when they reproduce. Ocean acidification occurs as seawater absorbs carbon dioxide. This changes carbonate chemistry and can make it more difficult for corals, shellfish, and some plankton to build or maintain calcium carbonate structures. Acidification does not turn the ocean into household acid. The change is smaller in ordinary pH terms, but marine organisms are sensitive to chemistry, and the shift is rapid compared with many past natural changes. The combined effect of warming, acidification, and oxygen loss is especially concerning because each can increase physiological stress. A marine animal may need more oxygen in warmer water, while less oxygen is available. At the same time, changing acidity may increase the energy required to build a shell or regulate internal chemistry. Energy spent coping with stress is energy not spent on growth, reproduction, movement, or immune defense. The animal may survive but produce fewer young. Once again, the warning can appear in reproduction before it appears in adult numbers. Climate change can also help invasive species expand into new regions, alter the timing of diseases, and increase wildfire risk in habitats already fragmented by development. This is why treating climate change as a separate environmental issue can be misleading. It modifies many of the other pressures. A species facing habitat loss may once have moved to a nearby cooler area. If the route has been cleared, movement is blocked. A species facing an invasive predator may once have recovered after a good breeding season. If drought reduces food, recovery becomes harder. A population exposed to pollution may be less able to survive heat. These interactions are sometimes described as synergistic, meaning the combined effect is greater than the sum of the individual effects. Two pressures do not simply add together. One can make the other more damaging. Imagine a seabird population whose nesting sites are disturbed by coastal development. The adults must travel farther to find quiet nesting space. At the same time, fishing reduces the availability of prey near the colony. Warmer seas move the remaining fish farther offshore. Plastic and chemical pollution affect feeding areas. A severe storm then destroys many nests. The storm may be described as the event that caused the breeding failure, but the population had already lost much of its margin for error. In a healthier system, birds might rebuild the next year. In the pressured system, another poor season may begin a sustained decline. This loss of margin is central to understanding why species approach the edge. Extinction often does not require every individual to be killed directly. It happens when deaths continue to exceed births, when populations become too isolated, or when random events become large relative to the number of individuals remaining. Small populations face what conservation biologists sometimes call an extinction vortex. Low numbers reduce genetic diversity and make it harder to find mates. Poor reproduction causes numbers to fall further. The smaller population becomes more vulnerable to random events, which then reduce it again. Once that process begins, removing the original threat may not be enough. A habitat can be protected, but the remaining individuals may still be too few or too scattered to recover without active help. This is why conservation sometimes involves captive breeding, moving individuals between populations, controlling predators, restoring food sources, or managing reproduction. Such work can look unusually involved because it is. By the time a species reaches a few dozen individuals, conservation may require knowing the family history, health, location, and romantic prospects of nearly every surviving member. Not all species receive that level of attention. Large mammals and birds are easier to notice and often easier to raise money for. A small snail, fungus, or beetle may be equally endangered while presenting certain difficulties for a dramatic fundraising poster. This produces gaps in knowledge and protection. The species most important to ecosystem processes may not be the ones people find attractive. Soil organisms, decomposers, pollinating insects, fungi, and plankton support systems on which larger animals depend. A forest without its famous mammals is visibly diminished. A forest without key fungi may continue standing for a while, making the problem harder to explain in a documentary trailer. Underlying all of these direct pressures are indirect drivers. Population growth can increase demand for land, food, energy, and materials, but population alone does not determine impact. Consumption differs greatly. One person may use far more resources than many others combined. Economic systems influence what is produced, where it is produced, and whether environmental costs are included in prices. A product may seem cheap because the cost of habitat loss, water pollution, or carbon emissions is paid elsewhere by communities, governments, future generations, or wildlife, none of whom are invited to the checkout. Subsidies can encourage harmful fishing, land clearing, fossil fuel use, or intensive production. They can also support restoration, cleaner energy, sustainable farming, and conservation. Laws and financial incentives determine which activities are rewarded. Global trade allows people to consume products made far from the environmental damage. Forest loss may happen on another continent. Fish may be caught in distant waters. Minerals may come from a region most buyers will never visit. This distance makes the connection between daily consumption and biodiversity harder to see. Governance matters as well. Protected areas can exist on maps while lacking staff, funding, community support, or enforcement. Environmental rules can be strong but poorly applied. Conservation can fail when local people bear the costs while receiving few benefits. Effective protection often works better when communities have secure rights, local knowledge is respected, and people gain from keeping ecosystems healthy. Conservation imposed without considering livelihoods can create conflict and may push harmful activities elsewhere. War, political instability, corruption, and poverty can increase pressure on wildlife, but the relationships are not simple. Conflict can reduce human activity in some places while increasing hunting, mining, logging, and institutional collapse in others. Poverty can force people to rely heavily on local resources, while wealth can drive far larger consumption through distant supply chains. There is no single villain turning one large wheel marked extinction. The pressures come from connected systems, and responsibility is unevenly distributed. That complexity can feel frustrating because it prevents one simple solution. We cannot protect biodiversity only by creating parks, only by reducing emissions, only by changing diets, only by controlling invasive species, or only by asking everyone to use a reusable bottle with exceptional enthusiasm. Each action addresses part of the problem. The most effective response depends on the species and place. An island bird may need invasive rats removed, a migratory fish may need a dam altered, a coral reef may need global warming reduced as well as local pollution controlled. A grassland plant may need traditional grazing or fire rather than complete exclusion of human activity. The shared requirement is to reduce the pressures faster than populations decline. That means identifying which driver is strongest, which interactions are accelerating damage, and where intervention has the greatest chance of success. To do that, scientists need more than a list of extinct species. They need measurements of population trends, habitat change, genetic health, ecosystem function, and future risk. They must separate temporary fluctuation from long-term decline and determine whether a population is recovering stable or moving toward a threshold from which recovery becomes much harder. That brings us to the practical challenge behind the larger question. If a mass extinction unfolds across many species, habitats, and timescales, how can anyone measure its progress? The answer requires fossils, field surveys, satellites, genetic samples, statistical models, and a surprising number of people willing to count animals that do not wish to be counted. Counting animals that do not wish to be counted is one of the basic difficulties of conservation science. A deer can walk behind a tree, a frog can stop calling when a researcher approaches, a fish can remain beneath water that is too dark to see through, a rare orchid can spend years underground before producing a flower, which is a remarkably effective way to avoid paperwork. Scientists therefore cannot measure an extinction in progress by making one visit to a habitat and writing down whatever happens to be visible. They need repeated observations, carefully designed surveys, statistical methods, historical records, and several different technologies. They also need enough patience to accept that not finding a species is not the same as proving that it is absent. The process usually begins with a baseline. Before researchers can say that a population has declined, they need some idea of its earlier condition. That baseline might come from old field surveys, museum collections, fishing records, hunting reports, photographs, scientific papers, local knowledge, indigenous knowledge, government documents, or observations made by volunteers. None of those sources is perfect. Old records may cover only places that were easy to reach. Collectors may have searched mainly for large or attractive species. Fishing records may describe what was caught rather than what was actually present. A museum might contain hundreds of specimens from one well-studied valley, and almost nothing from the surrounding region. Even imperfect records can become valuable when combined. A bird specimen collected in 1890 shows that the species lived in a particular place at that time. A notebook may reveal that it was once described as common. Later surveys can show when observations became less frequent. Satellite images can reveal whether its habitat was cleared. Modern genetic samples may show whether the surviving populations have become isolated. A baseline is not always a single number. It can be a map, a list of occupied sites, an estimate of population size, or a description of ecosystem condition. Scientists may ask how many individuals exist, how much land they occupy, how quickly the population is changing, and whether separate groups remain connected. The simplest method is a direct count. Researchers may count seals resting on a beach, birds nesting on a cliff, trees inside a plot, or animals crossing a migration route. If nearly every individual can be seen, the result can be quite accurate. Direct counts become harder when animals are small, mobile, hidden, nocturnal, widely scattered, or living in deep water. Counting every mouse in a forest would require cooperation from the mice, and they have so far shown little interest in a national census. Scientists often use samples instead. They count individuals within selected areas and use those results to estimate the wider population. The design of the sample matters enormously. Surveying only beside roads may miss species that avoid traffic. Visiting only in good weather may miss animals that are active during rain. Searching during the wrong season may produce a perfectly conducted survey of an empty breeding ground. Researchers try to choose sites that represent the full habitat and repeat the same method over time. If the survey changes each year, it becomes difficult to tell whether the population changed or the observers simply became better at finding it. Consistency does not mean every survey must remain frozen forever. New technology may improve detection. Habitats may shift, forcing survey areas to move. Scientists can adjust methods, but they must record those adjustments and account for them during analysis. One of the most important ideas in wildlife monitoring is detection probability. An animal may be present without being detected. It might remain silent, hide, move outside the observer's view, or simply be overlooked. A survey that records 0 individuals therefore does not necessarily describe an empty site. Occupancy models were developed to deal with this problem. Researchers visit the same location several times and record whether the species is detected during each visit. By comparing the pattern of detections and non-detections, the model estimates both the probability that the species occupies the site and the probability that observers will detect it when it is present. Early applications demonstrated that observed occupancy can substantially underestimate true occupancy when detection is imperfect. Imagine that a frog is heard during only half of the nights when it is actually beside a pond. A single quiet visit could lead someone to conclude that the frog is absent. 5 visits give a better chance of hearing it at least once. The statistical model uses those repeated visits to estimate how many apparently empty ponds may really contain unusually discrete frogs. Detection probability can change with weather, time of day, habitat, observer experience, and animal behavior. Birds may sing more during breeding season. Reptiles may be easier to see on warm mornings. Whales may remain submerged for long periods. A shy mammal may avoid a camera after noticing its scent. Scientists include these factors in their models when enough information is available. This is why field notes contain details that may appear excessive. Temperature, rain, wind, start time, equipment, observer identity, and survey duration can all influence the result. When researchers need an estimate of population size rather than simple presence, they may use capture-mark-recapture methods. Animals are captured, given a harmless identifying mark, and released. Later surveys record how many marked individuals are found again. Suppose researchers capture 100 turtles, mark them, and release them. During a later survey, they catch another 100 turtles, but only 10 are marked. That suggests the original marked group represented a relatively small share of the total population. If 50 are marked, the total population is probably smaller. Real calculations are more complicated because animals can move, die, lose marks, avoid traps, or become easier to capture after learning that traps occasionally contain food. Researchers use models that account for survival, movement, detection, and changes in the population between surveys. Modern marks include leg bands, tags, tiny implanted chips, photographs of natural patterns, and satellite transmitters. Whale sharks can be recognized from their spots. Tigers can be identified by stripe patterns. Some whales are known from the shape of their tails. Nature has supplied several species with their own identification cards, although it neglected to include addresses. Tracking devices reveal more than population size. They show migration routes, feeding areas, breeding sites, and places where animals die. A tagged bird may connect wetlands in several countries. A sea turtle may cross an ocean and return to one beach. A large mammal may leave a protected area each dry season because the reserve does not contain enough water. Those movements help scientists measure whether conservation boundaries match the needs of the species. A protected nesting site offers limited help if the feeding area is unprotected. A safe summer habitat cannot support a migratory animal whose winter habitat has disappeared. Camera traps have become another common tool. These are motion-triggered cameras placed along trails, near water, or in other places animals are likely to visit. They can operate day and night for weeks or months without a person standing nearby. The images reveal species that are difficult to observe directly. They can record nocturnal mammals, shy predators, and animals living in dense forest. If individuals have distinctive markings, photographs may also support population estimates. Camera traps are not neutral windows into the forest. Their position affects what they record. A camera placed on a wide trail may detect large mammals, while missing small animals moving through vegetation. Some species investigate cameras, others avoid them. Rain, leaves, livestock, and enthusiastic branches can trigger thousands of photographs containing no identifiable wildlife. Comparisons with other survey methods show that camera traps are highly useful, but not equally effective for every species or purpose. Their greatest value often comes from combining long operating periods with standardized placement and statistical treatment of detection. The resulting image collections can be enormous. A single project may collect millions of photographs. Human experts can identify the animals, but doing so requires a great deal of time and a willingness to examine many pictures of empty paths. Machine learning systems now assist by sorting images and suggesting species identifications. These systems can process data quickly, but they depend on good training material. An algorithm trained mostly on clear daytime photographs may struggle with a blurred animal at night. Rare species are especially difficult because there are fewer examples available for training. Automated identification therefore still needs quality checks. A computer that confidently labels a shadow as a leopard has not discovered a new population. It has discovered the importance of reviewing computer results. Some ecosystems are easier to hear than to see. Birds sing, frogs call, insects buzz, bats produce ultrasonic signals, whales communicate underwater, and many fish create sounds. Passive acoustic monitoring uses recording devices to collect those sounds over long periods. A recorder can remain in a forest through the night, operate during storms, and return data from times when fieldworkers would rather be asleep. In the ocean, hydrophones listen beneath the surface. Ultrasonic detectors record bats that human ears cannot hear. Researchers can search recordings for the calls of particular species. The timing and frequency of calls may reveal occupancy, migration, breeding activity, or responses to disturbance. Recordings also preserve the wider soundscape, allowing later researchers to ask questions that were not considered when the equipment was installed. The challenge is that sound does not translate directly into animal numbers. One loud frog can call repeatedly, while several quiet frogs remain silent. Wind, rain, insects, traffic, aircraft and machinery can hide biological sounds. An automated detector may confuse similar species or mistake a squeaking branch for a bird. Studies have found that acoustic indices can describe aspects of biological sound diversity in some terrestrial settings, while performing less reliably in others, including certain marine recordings. The method works best when researchers understand which organisms are producing the sound and combine acoustic results with observations from other sources. A quiet forest is not automatically an empty forest. It may have been recorded at the wrong time. The animals may communicate outside the recorder's frequency range. A healthy community of plants, fungi, and silent invertebrates may produce very little sound. A noisy habitat is not automatically diverse either. One abundant insect can dominate the recording, and nearby machinery can provide an impressive amount of acoustic activity without contributing much to biodiversity. Environmental DNA offers a different way to detect organisms. Animals, plants, fungi, and microbes leave genetic material in water, soil, air, sediments, and other surroundings. Fish shed cells and waste into rivers. Mammals leave hair and saliva. Plants release pollen. Tiny fragments remain after the organism has moved away. Scientists collect an environmental sample, extract the DNA, and search for genetic sequences linked to particular species. A water sample may reveal fish that were never caught or seen. Soil can contain traces of mammals, plants, fungi, and invertebrates. Air filters can collect genetic material from nearby organisms. Metabarcoding allows researchers to search for many groups at once. Instead of asking only whether one target species is present, they can compare the sequences in a sample with reference databases and build a broader picture of the community. The method can be extremely sensitive, non-invasive, and useful in places where traditional surveys are expensive or damaging. It also has limits. DNA can move away from its source, persist after an organism has left, degrade at different rates, or fail to appear in the sample even when the species is nearby. Laboratory procedures and data processing choices can create missed detections or false signals. Research has shown that detection errors can arise during water collection, filtration, amplification, and sequence analysis, which is why repeated samples and careful controls are necessary. A positive DNA result usually means that material from the species was present in the sampled environment. It does not always reveal how many individuals produced it. A large fish and several small fish may leave different amounts of DNA. Water flow can carry genetic material downstream. Temperature, sunlight, microbes, and chemistry affect how long it remains detectable. Scientists are developing methods to estimate abundance from DNA concentration, but the relationship varies between organisms and environments. For now, environmental DNA is often strongest as a tool for detection, community comparison, and early discovery of invasive or rare species. Genetic analysis can also measure diversity within a species. Researchers collect tissue, hair, feathers, droppings, or other material and examine how much genetic variation exists among individuals and populations. A population with low genetic diversity may have experienced a bottleneck, long isolation, or generations of inbreeding. Low diversity does not guarantee extinction. Some species survive with limited variation. It can, however, reduce the range of possible responses to disease and environmental change. Genetic information can reveal movement that field observations miss. Two animal groups separated by a road may still exchange enough individuals to remain connected. Populations that appear close together on a map may be genetically isolated because a mountain, dam, current, or behaviour prevents breeding. This allows conservationists to identify corridors, decide where crossings are needed, and sometimes move individuals to restore gene flow. It also helps determine whether two similar-looking populations belong to the same species, subspecies, or separate evolutionary lineages. Taxonomy may sound like a quiet activity involving labels and cupboards, but it changes extinction measurement. If one widespread species is later recognised as 3 separate species, each new species may occupy a smaller range and face a higher risk. If 2 named species are combined, the apparent number of threatened species may fall without any animal recovering. Scientists must therefore distinguish genuine biological change from changes in knowledge. A species may move into a higher risk category because its population declined, or because new information revealed that the earlier estimate was too optimistic. The conservation result may look similar, but the historical trend is different. The IUCN Red List provides a standardized system for evaluating global extinction risk. Assessors use 5 main criteria based on population reduction, geographic range, small population size and decline, very small or restricted populations, and quantitative estimates of extinction probability. The assessment draws on information from across the species' global range, rather than relying on one local population. The categories include least concern, near threatened, vulnerable, endangered, critically endangered, extinct in the wild, and extinct. Data deficient is used when available information is not sufficient for a proper assessment. Data deficient does not mean safe. It means the evidence is inadequate. Some poorly known species may be common, others may be close to extinction. The category is an honest admission that the measurement cannot yet support a stronger conclusion. Assessors examine the extent of occurrence, which describes the broad outer area containing the species, and the area of occupancy, which describes the smaller area actually inhabited within that range. A mountain animal may occur inside a large geographic boundary while using only a few isolated slopes. They also consider the number of mature individuals. Young animals are important, but extinction risk depends heavily on whether enough breeding individuals remain. A population containing many juveniles and few adults faces a different future from one with the same total number arranged across healthy age groups. Rates of decline are assessed across time periods linked to the species' generation length. 3 generations for a mouse may pass quickly. 3 generations for a whale can cover many decades. Using generation length helps the criteria reflect the biology of the organism rather than forcing every species onto the same clock. Quantitative models can extend those measurements into the future. Population viability analysis uses information about births, deaths, age structure, reproduction, environmental variation, catastrophes, and management actions to explore the risk of severe decline or extinction. Researchers can run many simulated futures. In one set, habitat continues shrinking. In another, protection improves. A third may include disease outbreaks, droughts, fires, or reduced hunting. The results show which factors have the greatest influence and which interventions are most likely to help. A population viability analysis is not a crystal ball. Its output depends on the structure of the model and the quality of the data. A species monitored for 4 years may behave differently over the next 50. Rare disasters are difficult to estimate because they are rare. Future climate and land use may not follow the assumed scenario. For these reasons, researchers have cautioned against treating population viability models as exact extinction calendars. Their strongest use is often comparing relative risks and management options rather than announcing that a species has precisely 27 years and 4 months remaining. This returns us to the question of time. A model might estimate a probability of extinction within 50 or 100 years. It may also estimate the chance of falling below a critical population size, sometimes called quasi-extinction. Quasi-extinction is useful because a species can become functionally lost before the final individual dies. A population may fall below the number needed for reliable reproduction or ecological function. Waiting to model only the literal last death would ignore the stage when intervention has the best chance of working. At larger scales, satellites help measure changes that no field team could observe everywhere. Earth-observing instruments track forest loss, fires, vegetation condition, surface water, ocean colour, temperature, snow, coastal change, and other features related to habitat. Satellites usually do not count individual beetles or identify every plant species from orbit. They measure the physical environment around them. When satellite observations are combined with field records, scientists can estimate habitat suitability, fragmentation, disturbance, and the movement of ecological boundaries. Long-running satellite programs are especially valuable because consistency over decades reveals trends. A single image shows where a forest is. A sequence of images shows whether it's being cleared, burned, regrown, or divided into smaller pieces. NASA describes biodiversity monitoring as a combination of repeated Earth observations and ground measurements, with remote sensing providing broad coverage that field surveys alone cannot achieve. Remote sensing can also identify changes in vegetation structure. Forest height and canopy density affect which animals can live there. Two landscapes may appear equally green in a basic image, while differing greatly in age, structure, and biological value. Ocean satellites measure temperature, colour, and other surface properties. Changes in ocean colour can indicate shifts in microscopic plant communities. Temperature records reveal marine heat waves that place coral reefs and other ecosystems under stress. The weakness of satellites is that broad patterns can hide local detail. A green area may be native forest, plantation, farmland, or invasive vegetation. A wetland can remain visible while pollution changes the organisms living in it. Ground surveys are needed to explain what the image means biologically. Museum collections provide another kind of a long-term measurement. Specimens preserve bodies, tissues, shells, bones, seeds, and sometimes parasites. They allow researchers to verify old identifications, study genetics, measure changes in body size, detect pollutants, and reconstruct former ranges. An old bird skin may contain chemical evidence of the environment in which it lived. A preserved fish can be compared with modern fish from the same river. Insects collected decades ago may reveal how seasonal emergence dates have changed. Collections also expose historical bias. Many were built during periods of colonial expansion, and sampling often concentrated in areas visited by European or North American institutions. Some regions and species are represented far better than others. Scientists must account for those patterns rather than assuming the collection is a balanced sample of the planet. Citizen science helps fill some gaps. Birdwatchers, divers, hikers, gardeners, photographers, fishers, and other volunteers contribute observations through organised surveys and public databases. Millions of people can cover far more ground than a small professional team. Volunteer data can reveal migrations, flowering times, invasive species, and changes in common populations. Repeated bird counts along standardised routes are especially valuable because participants follow consistent methods over many years. Less structured observations also matter. A photograph with a confirmed location and date can document a species far outside its known range. Many such records can help map distributions. The Global Biodiversity Information Facility brings together occurrence records from museums, researchers, monitoring projects, camera traps, and citizen scientists. Those records document where and when organisms have been observed and can support range mapping and red list measurements. The database also warns, through the pattern of its own records, that observations are uneven across geography and species groups. Popular animals near populated areas receive many observations. Small organisms in remote regions receive fewer. An apparent gap on a map may mean the species is absent, or it may mean no one looked there with the right method. Scientists try to correct for sampling effort. 10,000 bird observations from a city cannot be compared directly with 10 records from a remote forest. The city may have more observers rather than more birds. Standardized monitoring remains essential. Researchers need to know not only where a species was recorded, but how long people searched, which route they followed, what equipment they used, and what they failed to detect. Absence data are surprisingly valuable. A list of sightings shows presence. A repeated survey that searched carefully and found nothing helps estimate absence. This is one reason a casual photograph and a structured field survey answer different questions. Confirming global extinction is the hardest absence problem of all. A species may be rare, hidden, seasonal, or living in an unexplored part of its former range. Some species have been rediscovered after decades without a confirmed record. The IUCN defines a species as extinct when there is no reasonable doubt that the last individual has died. This requires surveys in known and expected habitat, conducted at appropriate times and across the historical range, with efforts suited to the organism's biology. Species that are probably gone but lack adequate confirmation may remain critically endangered with a possibly extinct flag. Declaring extinction too early can end searches and protection. Declaring it too late can make official statistics underestimate loss. Conservationists must balance the risk of wasting effort against the much greater problem of abandoning a species that still survives. Searches are designed around the species. A nocturnal bird should not be surveyed only at midday. A plant that flowers once every several years may require repeated seasonal visits. An aquatic species may need nets, visual surveys, acoustic equipment, and environmental DNA. Local knowledge can guide those searches. People who live or work in the habitat may recognize calls, tracks, seasonal patterns, or recent changes unknown to outside researchers. Such information needs careful verification, but dismissing it can mean overlooking decades of observation. Measuring a global mass extinction requires another step. Scientists must compare losses among thousands of species and across very different timescales. Modern records may describe individual populations over years. Fossil data often combine events occurring across thousands or millions of years. The fossil record is incomplete and uneven. Organisms with hard shells or bones are more likely to be preserved. Marine sediments often provide better records than tropical forests. Some periods and regions contain abundant exposed rock, while others leave fewer accessible deposits. Research has shown that the quantity and distribution of available sedimentary rock can influence apparent patterns of ancient diversity and extinction. Paleontologists use statistical corrections, compare multiple regions, and study groups with relatively good fossil records. They estimate how long species normally persisted and identify intervals when disappearance rates rose sharply. Modern extinction rates can then be compared with background estimates, but the calculation contains choices. Researchers must decide which species groups to include, how to treat possibly extinct species, which background rate to use, and how to handle species that have not been formally assessed. This explains why different studies produce different numbers without necessarily producing opposite conclusions. One estimate may use only confirmed vertebrate extinctions. Another may include species considered probably extinct. A third may estimate losses from habitat change. The methods answer related but not identical questions. Indirect estimates require particular care. Scientists sometimes use the relationship between habitat area and species richness to estimate how many species may eventually disappear after habitat loss. This can identify serious risk, but the timing and final number depend on how species respond, whether they survive in fragments, and whether habitat is restored. A species can remain for years after losing much of its habitat, creating an extinction debt. Another may adapt to altered landscapes better than expected. Models are useful when their assumptions are clear, not when their results are treated as a simple count of invisible extinctions. No one instrument can measure the entire crisis. Field counts provide direct information but cover limited areas. Satellites cover the globe but usually cannot identify individuals. Environmental DNA detects hidden organisms but may not estimate abundance. Camera traps work well for certain animals and poorly for others. Acoustic recorders hear vocal species but miss silent ones. Museum collections extend the timeline but reflect historical sampling bias. The strongest assessments combine them. A satellite may reveal that a wetland is shrinking. Field surveys show that bird numbers are falling. Acoustic recorders detect fewer frog calls. Water samples reveal the loss of several fish species. Genetic analysis shows that remaining populations are isolated. A population model estimates that restoring water flow would improve survival. Each result supports the others. Together, they turn scattered observations into evidence of a system moving toward or away from danger. Scientists also need measurements of recovery. A population increasing from 50 to 100 individuals has improved, but it may remain highly threatened. A species may become more numerous while occupying only a fraction of its former range. Extinction risk and full ecological recovery are not the same thing. The IUCN Green Status Framework was created to assess how close a species is to being fully recovered across its natural range, and whether conservation actions have contributed to improvement. This complements risk categories by asking not only how close a species is to disappearing, but how much of its former ecological presence has been restored. That difference matters because success should not mean keeping a species permanently alive in one fenced reserve or breeding centre. The broader goal is usually a secure, functioning species occupying suitable parts of its natural range. An animal that survives only in captivity is protected from immediate global extinction, but it is no longer performing its role in the wild. Reintroduction requires habitat, food, social behaviour, disease management, genetic planning, and control of the threat that caused the original decline. Measurement, therefore, does more than record bad news. It tells conservationists where to act, whether an intervention works, and when a strategy needs to change. If a population increases after hunting is reduced, enforcement may be working. If it continues falling, habitat loss, disease, or another pressure may be more important. If young animals survive but adults die near roads, wildlife crossings or traffic controls may offer greater benefit than additional breeding programs. Good monitoring can prevent resources from being spent on actions that sound helpful, but fail to address the main cause. It can also identify success before the recovery is obvious to the public. There will always be uncertainty. Some species remain unstudied. Some regions lack long-term surveys. Wars, limited funding, difficult terrain, and shortages of trained experts create gaps. Taxonomy changes. Equipment fails. Animals move. Data can be misidentified, duplicated, or stored where other researchers cannot access it. Uncertainty is not a reason to avoid decisions, it is part of the decision. Scientists can describe a range of likely outcomes, identify which unknowns matter most, and update conclusions as evidence improves. A smoke detector does not need to measure every molecule in the room before becoming useful. Biodiversity monitoring works in a similar way. It cannot observe every organism, but it can detect patterns that are too large and consistent to dismiss. The current measurements show losses, but they also show places where populations stabilise or return. That is the practical value of measuring an extinction in progress. The same tools that record decline can reveal whether the decline has slowed. The next question is therefore not only how accurately scientists can follow species toward the edge, it is whether human action can change the direction once the warning is clear, and whether conservation can operate quickly enough to matter before small populations become recoveries that no model can reasonably expect. A recovery that no model can reasonably expect is not reached at one fixed population size. Some species have returned from fewer than 100 individuals. Others have continued declining despite thousands remaining. The difference usually lies in their biology, the condition of their habitat, the threat still acting on them, and whether conservation begins early enough. That gives us a practical answer to the question of whether humanity can slow the clock. Yes, it can. Conservation has prevented extinctions, restored habitats, rebuilt populations, and reduced specific threats in many parts of the world. The more difficult answer is that successful projects are still too limited to outweigh the total scale of habitat destruction, exploitation, pollution, invasive species, and climate change. In other words, the brakes work. The problem is that the accelerator is still being pressed. This is not merely an encouraging collection of isolated stories. A large global analysis published in 2024 examined 186 studies containing 665 comparisons between conservation action and situations in which no comparable action occurred. The interventions included habitat protection, restoration, invasive species control, sustainable management, and direct work with threatened species. In roughly 2/3 of the cases, conservation improved biodiversity, or slowed its decline compared with doing nothing. The actions did not always return ecosystems to their original condition, but they usually produced a better outcome than the alternative. That distinction is important. A conservation project can succeed even when the population continues to fall. Suppose a seabird colony would have declined by 10% each year without action, but declines by 2% after fishing practices are changed. The species is still in trouble, yet the intervention has bought time. That time can be used to address other threats, improve breeding success, or expand protection. People sometimes judge conservation only by complete recovery. If the species is not abundant again, the work is described as a failure. That is rather like calling a fire brigade unsuccessful because the repaired house does not immediately look new. Preventing total destruction is already useful. Rebuilding comes afterward. The strongest conservation usually begins with removing or reducing the main cause of decline. If animals are being hunted faster than they reproduce, hunting pressure must fall. If a wetland is drying because water is being diverted, the flow must be restored. If rats are eating the eggs of island birds, controlling those rats may matter more than planting trees or installing informative signs explaining that egg theft is discouraged. This sounds obvious, but conservation projects do not always target the strongest threat. Sometimes the main cause is politically difficult, expensive, or located outside the protected area. It may be easier to build nesting boxes than to change the industry removing the animal's food. It may be easier to plant seedlings than to stop the activity that repeatedly destroys the forest. A nesting box can still help. Planting can still help. The problem comes when the visible action replaces the necessary one. Research on threatened terrestrial species has found large gaps between known threats and documented responses. One 2024 assessment concluded that many species facing habitat loss, invasive species, or international trade lacked adequate intervention. It found that 58% of the threatened terrestrial species examined had conservation measures that were notably insufficient or absent. It also found that most species threatened by habitat loss did not have enough of their habitat represented inside protected areas to meet the study's threshold. Simply placing a species inside a protected boundary does not guarantee recovery. The protected area may cover only a small part of its range. It may exclude feeding grounds, migration routes, or seasonal water sources. Management may exist on paper but lack staff, funding, enforcement, or cooperation from nearby communities. A park surrounded by clearing, roads, and pollution can become an island. Species trapped inside may lose genetic connections with other populations. Animals may leave the boundary to find food and encounter hunting or traffic. Rivers carry whatever happens upstream through the reserve, apparently unaware that they have crossed into a legally protected zone. Effective protection therefore requires more than reaching a percentage on a map. The protected places must contain important habitats, represent different ecosystems, remain connected, and be managed in ways that produce real conservation outcomes. The Global Biodiversity Framework, adopted under the Convention on Biological Diversity, calls for at least 30% of land, inland water, coastal areas, and oceans to be effectively conserved by 2030. It also says those areas should be ecologically representative, connected, and equitably governed, with recognition of indigenous and traditional territories and respect for the rights of indigenous peoples and local communities. The word effectively does a great deal of work in that target. A protected area that allows destructive extraction, has no management plan, or exists only because the location was not wanted for anything else, may contribute to the official total without protecting the species most at risk. Choosing the right places matters as much as choosing enough places. A small wetland used by thousands of migrating birds may protect more ecological value than a much larger area with few threatened species. A narrow forest corridor may connect two large reserves and prevent populations from becoming isolated. A short stretch of river may contain the only spawning habitat used by a fish. Conservation planning therefore tries to identify areas with high numbers of unique species, breeding sites, migration routes, climate refuges, and ecological functions that cannot easily be replaced. Protecting every square kilometre equally would be simple, but biology has not arranged itself for administrative convenience. Connections between habitats are becoming more important as the climate changes. A protected area designed around present temperatures may become unsuitable for some of its species in future decades. If those species can move through connected landscapes, they may reach cooler or wetter habitat. If the reserve is surrounded by roads, farms, walls and cities, the legal protection may preserve a location, while the climate that made it useful moves elsewhere. Wildlife corridors are one response. These may be continuous strips of natural habitat, restored riverbanks, forest bridges over roads, tunnels beneath highways, hedgerows through farmland, or carefully managed areas where species can travel without facing excessive danger. Corridors are not suitable for every situation. They can also assist disease, fire, invasive species, or predators. Their design must match the species and landscape. A tunnel useful to a badger will not be especially impressive to a butterfly, although the butterfly may appreciate the effort. For isolated populations, conservationists may move individuals directly. Translocation can restore a species to part of its former range, strengthen a small population, or increase genetic diversity. The animals may come from another wild population or from a breeding program. Moving wildlife is not as simple as opening a box and wishing everyone well. The release site must contain suitable food, shelter, breeding conditions, and protection from the original threat. Animals may need health screening to prevent disease. Their genetics must be considered. Their age and social behaviour can affect survival. Some species need to learn how to find food or avoid predators. Captive-bred animals may require training before release, which can involve presenting safe versions of predators or teaching them to recognise natural foods. Conservation biology occasionally resembles an unusually demanding school whose students may be eaten if the lesson plan is poor. Captive breeding is most useful when it supports recovery in the wild, rather than becoming permanent storage for a species that has nowhere safe to return. Zoos, aquariums, botanical gardens, seed banks, and specialised breeding centres can protect genetic material and produce individuals for reintroduction. They can also buy time while habitat is restored. That time is not unlimited. Small captive populations can lose genetic diversity, develop health problems, or adapt to captivity. A species bred for many generations under human care may become less suited to wild conditions. Careful breeding records are used to reduce inbreeding and preserve variation. Genetic diversity matters because it helps populations respond to disease and environmental change. A global analysis covering 628 species found evidence that genetic diversity is being lost within populations, particularly where land use change, disease, harvesting, and other pressures are strong. The same study found that management actions such as restoring connectivity, improving population growth, and introducing individuals from other populations can help maintain or increase genetic diversity. This is one reason saving several separated populations can be better than keeping one large group in one place. Different populations may contain different genetic traits. One may tolerate heat better. Another may resist a disease. Another may reproduce at a slightly different time. Those differences can become important as conditions change. Combining every surviving individual into one population may increase numbers while reducing the geographic and genetic variety that once protected the species from regional disasters. Habitat restoration is another major tool, but restoration is often misunderstood. It does not mean making a place green. A golf course is green. So is a field of one crop. Neither automatically replaces a wetland, forest, grassland or reef. Restoration begins by deciding which ecological functions need to return. That may involve restoring water flow, removing contamination, controlling invasive plants, rebuilding soil, reintroducing grazing, allowing natural fire, planting native species, or removing barriers from rivers. Sometimes the best action is active construction. At other times, it is stopping the disturbance and allowing natural recovery. A forest near surviving woodland may regenerate when grazing and repeated clearing end. A heavily damaged mine site may require extensive work before plants can establish. The Global Biodiversity Framework calls for at least 30% of degraded terrestrial, inland water, coastal, and marine ecosystems to be under effective restoration by 2030. The goal is not merely to begin projects, but to improve ecological integrity, functions, services, and connections. A restored ecosystem does not need to become an exact copy of a historical photograph. Climate, surrounding land use, and species distributions may already have changed. Some extinct local species cannot be returned. Restoration may instead aim for a healthy, diverse, self-sustaining system suited to present and expected future conditions. Poorly designed restoration can cause new problems. Planting trees in a natural grassland may reduce native biodiversity. Choosing fast-growing non-native trees may create a plantation with little habitat value. Restoring a wetland without addressing polluted water upstream may produce a very expensive pond full of algae. Success must be measured over time. Young trees planted during a public event may look impressive in the first photograph. The more useful question is, how many are alive 10 or 20 years later, whether native plants have returned beneath them, whether animals use the habitat, and whether the system can reproduce without continual planting. Restoration cannot replace protection of intact ecosystems. Rebuilding a mature forest is slower, more uncertain, and usually more expensive than preventing its destruction. Some ecological features take centuries to develop. Old trees, deep soils, complex fungal networks, and stable relationships between species cannot be installed quickly. An intact ecosystem should not be cleared on the promise that another will be created elsewhere. Ecological compensation can sometimes address limited damage, but it becomes unreliable when the habitat is unique, ancient, or difficult to reproduce. Replacing a 1,000-year-old woodland with seedlings is not a direct exchange. It is a long experiment conducted after the original has been removed. Invasive species control has produced some of the clearest conservation successes, especially on islands. Removing introduced rats, cats, goats, pigs, and other animals can allow native birds, reptiles, plants, and invertebrates to recover. A global assessment of invasive vertebrate eradications found that island restoration projects have produced benefits for native biodiversity and can support later actions such as reforestation and wildlife reintroduction. Eradication is possible when the area is limited, reinvasion can be prevented, and every remaining individual of the invasive population can be reached. On continents, complete removal is much harder, so management may focus on preventing arrival, slowing spread, or protecting important sites. Prevention is usually cheaper and safer than control after establishment. That means inspecting cargo, managing ballast water, regulating high-risk trade, monitoring ports, and responding quickly to new detections. Rapid response can feel excessive when only a few unfamiliar insects or plants have appeared. It becomes much less excessive after those few organisms have spread across an entire country and developed a firm position on the landscape. Direct exploitation can be reduced through quotas, seasonal restrictions, protected breeding areas, trade controls, and enforcement. These measures work best when they're based on reliable data and supported by the people whose livelihoods depend on the resource. A fishing limit that ignores food security or local knowledge may fail because people cannot reasonably follow it. A rule designed with fishing communities can protect spawning periods, reduce damaging gear, and maintain catches over the longer term. Sustainable use is not the same as no use. It means harvesting at a rate and in a way that allows populations and ecosystems to continue. In some places, well-managed hunting or fishing provides incentives to maintain habitat and prevent illegal exploitation. In others, even low levels of removal may be unsafe because the population is already too small. The correct approach depends on reproduction, mortality, population size, and enforcement. A fast-growing fish can tolerate a different harvest rate from a whale that produces one calf after a long pregnancy and then spends years raising it. Pollution control can lead to relatively rapid improvements when the source is clear. Sewage treatment can restore oxygen levels in rivers. Restrictions on harmful chemicals can allow birds and insects to recover. Reducing fertilizer runoff can limit algal blooms. Changes in fishing equipment can reduce bycatch. These actions often require rules applied across industries rather than voluntary changes by a few careful individuals. One farmer reducing nutrient loss helps, but a damaged river receives water from the entire catchment. One quiet ship helps, but migratory whales encounter many vessels. Policy determines the background conditions in which individual choices operate. Products, energy, transport, food, and waste systems influence biodiversity long before a consumer sees the final item. This is why changing financial incentives matters. Governments may support activities that increase land clearing, overfishing, pollution, or fossil fuel use, sometimes because the policy was created for another goal, and its environmental effects were not fully considered. The Global Biodiversity Framework calls for harmful incentives and subsidies to be identified, reformed, phased out or eliminated, with reductions of at least $500 billion per year by 2030, while positive incentives for conservation are expanded. A conservation department can spend money restoring wetlands, while another part of government pays for activities that drain them. This is not an efficient arrangement, although it does create employment for people repeatedly repairing the same problem. Redirecting finance can support farmers who maintain hedgerows, landowners who protect habitat, fishers who adopt less damaging equipment, and communities that conserve forests. Payments must be designed carefully, so they produce real ecological outcomes rather than rewards for actions that would have happened anyway. Private companies also affect biodiversity through supply chains, investment, infrastructure, and land use. A business may not own a forest, but may purchase products associated with clearing. It may not operate a mine, but may finance one. Tracing those connections allows companies, regulators, investors, and consumers to identify where damage occurs. Corporate promises are useful only when they contain measurable targets, transparent reporting, and consequences for failure. A statement announcing respect for nature does not protect a single hectare by itself. Forest animals remain surprisingly resistant to public relations language. Climate change cannot be separated from conservation. Protected habitats, restored rivers, and invasive species control may all fail if temperatures and extremes move beyond what species can tolerate. Reducing greenhouse gas emissions is therefore one of the broadest actions available for lowering future extinction risk. Conservation can also help species adapt to unavoidable change by protecting climate refuges, connecting habitats, restoring coastal systems, and reducing other pressures. The Convention on Biological Diversity's climate target calls for reducing the effects of climate change and ocean acidification on biodiversity while increasing ecological resilience through mitigation, adaptation, and disaster risk measures. nature-based climate actions need careful design. Restoring peatlands, mangroves, forests and seagrass can store carbon while supporting biodiversity. Planting one fast-growing tree species across a natural grassland may store some carbon while damaging the ecosystem that already exists. Climate and biodiversity goals overlap, but they are not automatically identical. A renewable energy project can reduce emissions and still harm wildlife if it is built across an important migration route. A dam may produce low-carbon electricity while disrupting a river. Planning must consider both goals rather than assuming anything labelled green is harmless. Local and indigenous communities are central to many successful conservation efforts because they often live in, use and manage areas with high biodiversity. Their knowledge can include seasonal cycles, animal behaviour, fire management, water, plants, and changes that are not recorded in formal scientific surveys. Recognising this knowledge does not mean treating every traditional practice as automatically sustainable, or every community as internally uniform. It means that conservation is more effective and more just when the rights, institutions, experience and priorities of the people who live with the ecosystem are included from the beginning. Protected areas created through forced removal or exclusion can cause serious harm and conflict. People may lose homes, food, sacred places or livelihoods in the name of protecting a landscape they helped maintain. Conservation that violates rights is not repaired simply by producing a good wildlife count. The current global framework explicitly links area-based conservation with equitable governance and recognition of indigenous and traditional territories. IUCN guidance also stresses the importance of recognising and supporting territories and areas conserved by indigenous peoples and local communities. Funding remains one of the largest practical limits. Monitoring species, buying land, restoring habitat, enforcing laws, removing invasive organisms, and supporting communities all require money over long periods. Short grants can launch a project, but ecosystems do not always arrange recovery within a 3-year funding cycle. A breeding program may need decades. A restored forest may require management across generations. Rangers, researchers, community organisations and local governments need reliable support after the opening ceremony has ended and the large ceremonial shovel has been returned. The Global Biodiversity Framework calls for at least $200 billion per year from all sources for biodiversity by 2030, including increased international finance for developing countries. The target reflects the gap between the scale of the agreed goals and the resources currently available to implement them. Money alone does not guarantee success. It can be poorly targeted, delayed, or spent without monitoring. Yet conservation without adequate finance relies on underpaid staff, volunteers, temporary projects and communities asked to carry global responsibilities without sufficient support. Priorities are unavoidable because resources are limited. Conservationists sometimes face decisions about which species, habitats or actions to fund first. This is uncomfortable because every threatened species has value, but ignoring priorities does not make scarcity disappear. One approach is to focus on species that can be saved through realistic action. Another protects places containing many threatened species. Some projects prioritize species with unique evolutionary histories or important ecological roles. Others act where the decline is fastest. Triage should not become an excuse for abandoning difficult species or allowing preventable threats to continue. A species may look expensive to save only because society continues funding the activity that endangers it. Early action is usually cheaper than emergency rescue. Protecting a population while it remains large requires less intensive work than rebuilding it from 12 individuals. Preventing an invasive species from arriving is easier than removing it from thousands of square kilometres. Keeping a forest standing is more reliable than recreating it. That gives humanity a strong reason to act before extinction risk becomes extreme. It also means conservation cannot focus only on famous species already close to disappearing. Common species, intact habitats, and ordinary ecological processes need protection before they become emergency cases. Individual choices have a role, but they should be kept in proportion. People can reduce waste, choose products with lower impacts, create habitat in gardens, avoid releasing pets, support conservation groups, and vote for policies that address the main drivers. A garden containing native plants can help local insects and birds. It cannot replace a tropical forest, an ocean food web, or national climate policy. Personal action is most effective when it supports wider institutional change rather than becoming a substitute for it. No one needs to achieve ecological perfection before participating. Modern life makes some environmental impact unavoidable. The useful question is not whether a person can live without affecting nature. It is which changes reduce the largest pressures and help create systems in which better choices become normal rather than difficult. Humanity can slow extinction because the drivers are not mysterious. We know that populations respond when hunting falls, habitats recover, pollution is removed, movement routes reopen, invasive predators are controlled, and breeding conditions improve. We also know that partial action may fail when the strongest pressure remains. The evidence does not support the idea that nature will recover automatically regardless of what we do, nor does it support the idea that every attempt is pointless. Conservation works, but it must operate at the scale and speed of the damage. A protected valley can save a plant. Restored water can return fish to a river. Removing rats can restore an island colony. Reducing emissions can lower risks across entire regions. Reforming laws and markets can prevent damage before a conservation team is required to repair it. The clock is not one machine with one switch. Each species has its own population trend, each ecosystem has its own limits, and each pressure moves at a different rate. Some losses are already permanent, others can still be prevented. Some populations need emergency intervention now, while others need ordinary protection so they never reach that point. Slowing the clock therefore means slowing thousands of separate declines at once. It means turning threatened populations into stable ones, stable populations into growing ones, and isolated habitats into connected systems. It means measuring success not by promises made, but by whether more species remain alive, reproduce successfully, and recover their ecological roles. If those efforts expand, the possible sixth mass extinction does not have to reach the scale of the previous 5. The geological record may eventually show a severe human-caused biodiversity crisis that was slowed before 3/4 of species disappeared. If the efforts remain too small, arrive too late, or are repeatedly overwhelmed by larger destructive systems, life will still continue. Earth has passed through mass extinction before. Microbes, plants, fungi, and animals would survive in some form, and evolution would eventually fill many empty roles. The comforting phrase is that life finds a way. The less comforting detail is that it may require millions of years, and the life that returns would not be the world we recognize now. Understanding that difference requires looking beyond the last individuals of threatened species and considering what happens after a large share of the living world is removed, which organisms are most likely to survive, and what kind of planet those survivors would begin to rebuild. The survivors would not begin by rebuilding the world that had just been lost. They would begin by staying alive. That distinction appears throughout the fossil record. Immediately after a major extinction, the first successful organisms are often not the ones that create the most complex ecosystems. They are the ones that can tolerate unstable temperatures, poor water quality, limited food, damaged soil, low oxygen, or repeated disturbance. They may grow quickly, reproduce early, travel far, eat several kinds of food, or survive in places that protect them from the worst conditions. In an ordinary ecosystem, specialization can be extremely effective. One animal may feed on one plant that no competitor can digest. One insect may reproduce only inside one type of flower. One reef animal may be perfectly adapted to a narrow range of temperature and water chemistry. Those relationships work well while the surrounding system remains reliable. During a mass extinction, reliability disappears. The specialist may still possess an excellent solution to a problem that no longer exists. The early survivors are sometimes called disaster taxa. This does not mean they caused the disaster or enjoyed it. It means they became unusually common in the simplified ecosystems that followed. They occupied space left by extinct species and tolerated conditions that prevented many competitors from returning. Some disaster taxa were already common before the crisis. Others had lived quietly in limited habitats and expanded only after the environment changed. Their success could be brief. Once stable, diverse ecosystems returned. The same traits that helped them during the emergency might no longer provide an advantage. After the end Permian extinction, environmental instability continued for millions of years. Warming, low oxygen, disrupted carbon cycles, and repeated ecological stress slowed the rebuilding of complex marine and terrestrial communities. Some groups diversified within the first few million years, but stable ecosystems with several trophic levels took much longer to reappear. A major synthesis estimated that broadly complex ecosystems were not restored until roughly 8 to 9 million years after the crisis, although newer work emphasizes that recovery differed among places and that some complex communities returned earlier than the old simple step-by-step model suggested. That tells us something important about the word recovery. It does not describe one event. Primary production may return first. Algae, plants, and photosynthetic microorganisms begin capturing energy again. Small herbivores and detritus feeders increase. Predators follow when enough prey becomes available. Habitat-forming organisms return where conditions allow. Species numbers rise. Food webs gain more connections. Populations become less unstable. Each of those changes can occur at a different speed. An ocean may begin producing large amounts of biological material while still containing a simplified and unreliable community. A forest may become green long before it develops old trees, deep soils, nesting cavities, fungi, large seed dispersers, and predators. The aftermath of the asteroid impact 66 million years ago shows this difference clearly. Evidence from inside the Chicxulub crater indicates that life returned to the basin within years, and a productive ecosystem became established within about 30,000 years. That is rapid by geological standards, though it remains slightly longer than most people allow for a garden to recover after forgetting to water it. Elsewhere in the oceans, however, community stability and carbon cycle function took much longer. A detailed record of marine plankton found about 1.8 million years of unusually unstable post-extinction communities before a more resilient arrangement appeared. Species richness remained low even after important ecosystem functions had recovered, showing that biological productivity, ecological stability, and full diversity are separate measurements. On land, the first visible recovery can also be misleading. Fern spores often become abundant in rock layers after severe disturbance because ferns can colonize open ground quickly. They do not need an intact forest canopy and can spread into damaged landscapes where slower growing plants have disappeared. A fern-covered landscape is alive and recovering, but it's not yet the forest that existed before. After the end Cretaceous extinction, plants and their insect herbivores recovered at different rates in different regions. Fossil leaves from Patagonia indicate that the diversity of insect-feeding relationships returned to its earlier level in about 4 million years, while comparable recovery in parts of Western North America took close to 9 million years. Geography, climate, surviving species, and chance all influence the result. This regional variation prevents us from describing a future mass extinction recovery with one global schedule. Some places would retain more species than others. Mountain valleys, deep water, caves, wetlands, high latitudes, and isolated habitats might provide refuge from particular threats. The same places could become traps if conditions continue changing and the survivors have nowhere else to move. Survival would depend on the cause of the crisis. A burrowing mammal may survive a short period of darkness and fire but struggle during centuries of drought. A freshwater turtle may endure a sudden collapse in land vegetation because it can eat varied food and survive long periods without feeding. Yet it may be unable to cope with a river that remains polluted and dammed for generations. There is no universal list of animals that always survive mass extinctions. Small size, fast reproduction, broad diets, wide ranges and flexible behaviour often help, but every event selects organisms differently. The end Cretaceous impact placed an immediate premium on surviving darkness, cold, and the failure of food chains based on fresh photosynthesis. New modelling of marine plankton suggests that darkness and body size related energy requirements helped shape which plankton survived. Small organisms and forms able to combine photosynthesis with feeding were better placed than larger specialists when light and food collapsed. The present crisis applies a different set of filters. Habitat conversion, harvesting, pollution, invasive species, disease, and climate change often act for decades or centuries rather than arriving as one brief impact winter. Species must survive not only an initial shock but continued pressure and shrinking space. Large animals are frequently vulnerable because they need more territory, occur at lower population densities, reproduce slowly, and are often targeted by hunting or fishing. Specialists are vulnerable when the one habitat or food source they require disappears. Species with narrow geographic ranges have fewer alternative populations if one region is damaged. Models of future mammal and bird losses predict that extinction would remove ecological strategies selectively rather than randomly. The surviving species pool would shift towards smaller, faster-lived, more fertile generalists, including many animals that eat insects and use several habitat types. That would not mean every rat, crow, or beetle wins, but it suggests a world with fewer large, slow-breeding specialists and more species capable of living around disturbance. Such a world might still look busy. City parks would contain birds, fields would contain insects, harbours would contain fish, roadsides would grow plants. The silence in the title would not be literal silence everywhere. The change would be found in what was missing and in how often the same adaptable organisms appeared. Unique local species could decline while widespread species expanded. A forest might retain many plants, but lose its largest fruit-eating animals. An island might remain green while its native birds were replaced by introduced birds, rodents, and insects. Two distant ecosystems might begin sharing more of the same successful species. Scientists call this biological homogenization, when communities become more similar. It is a useful risk to consider, but current evidence does not show one universal pattern across every habitat and pressure. A large 2025 synthesis found that human pressures generally reduce local diversity and alter community composition, while the degree and direction of homogenization vary among ecosystems, organism groups, and types of disturbance. The future would not be one simple, global collection of pigeons and weeds. It would be a complicated mixture of local losses, new arrivals, altered communities, and a few persistent specialists. The disappearance of large animals would change more than the appearance of the landscape. Large herbivores move seeds, open vegetation, create trails, dig waterholes, and transport nutrients. Predators influence where prey feed and how many survive. Scavengers remove carcasses. Large fish and marine mammals move nutrients through the ocean. If those animals disappear, their functions are not automatically transferred to smaller species. 10,000 mice do not become one elephant simply because the total weight is similar. They eat different foods, travel different distances, disturb soil differently, and place seeds in different locations. Plants would respond over generations. Trees with large fruits may lose the animals capable of swallowing and dispersing their seeds. Seedlings could become concentrated near parent trees, where competition, disease, and predators are often stronger. Forest composition might shift toward plants spread by wind, water, small animals, or humans. A study examining palm fruits and mammal seed dispersers projected that continued loss of large fruit-eating mammals would reduce the body size of available dispersers and weaken the ecological relationship supporting some of the largest fruits. The palms would not all vanish immediately, because adult trees can live for a long time. The change would appear later through reduced recruitment and altered forest structure. This delay means the world after an extinction could continue losing functions even after the original pressure slows. Old trees remain after their dispersers disappear. Coral skeletons remain after living reefs die. Soil stores seeds from plants no longer reproducing successfully. Adult animals survive after breeding has failed. Recovery begins only when births and colonization consistently exceed further losses. If the climate continues warming, habitats continue shrinking, and new species keep arriving through trade, there may be no stable post-extinction period. The ecosystem is not recovering from a completed event. It is trying to reorganize while the conditions continue changing. Previous mass extinctions ended because the main physical disturbance eventually weakened. Volcanic eruptions stopped. The atmosphere and oceans gradually changed. Dust cleared after the asteroid impact. Habitable conditions expanded. A human-driven extinction has an unusual feature. The species causing much of the pressure can observe it, measure it, and choose whether to continue. That creates the possibility of slowing the event, but it also means there may be no natural stopping point if land conversion, exploitation, pollution, and emissions continue. Suppose those pressures were greatly reduced. The first response would not be the sudden appearance of lost species. Existing populations would have more room to reproduce. Rivers would carry less pollution. Forest edges would regenerate. Fish would survive long enough to reach breeding age. Animals would move through restored corridors. Some populations would increase within years. Others would require decades. Ecosystem functions might recover before species richness. This occurred after ancient extinctions and can happen in modern restoration. A few species may provide pollination, decomposition, or primary production, while the wider community remains simplified. That function is valuable, but it should not be mistaken for complete recovery. New species would eventually evolve. Surviving populations become separated, adapt to different conditions, and accumulate genetic differences. Over long periods, one lineage can split into several species. Empty ecological roles create opportunities for expansion and experimentation. After the end Cretaceous extinction removed the non-avian dinosaurs and many other groups, surviving vertebrates diversified into newly available ways of life. Terrestrial vertebrate richness rose sharply in the earliest Paleogene, and major groups of modern marine fishes also began important radiations near the extinction boundary and during the first 10 million years that followed. Mammals are the familiar example, but their story is not simply that tiny, identical creatures waited for dinosaurs to leave and then immediately turned into horses, whales, and accountants. Mammals were already ecologically varied before the impact. Some lineages survived, others vanished, and the survivors expanded into a larger range of body sizes, diets, and habitats over millions of years. Birds followed their own paths. Snakes, frogs, fishes, insects, and plants also diversified. New communities formed through interactions among survivors, not through a planned replacement of every extinct species. Evolution does not keep job descriptions. When a large predator disappears, a future predator may occupy a similar position, but it will have a different ancestry, anatomy, behavior, and effect on its environment. A new reef builder may create habitat, but not in the exact shape or location of the old reef. Similar functions can return without restoring the former world. Some empty roles may remain unfilled for a long time. Recovery after the end Permian crisis included repeated environmental setbacks, and complex communities were assembled unevenly. The earliest successful organisms were not simply waiting to mature into a complete ecosystem. They competed, spread, disappeared, and were replaced while the physical environment continued to shift. Freshwater records from Australia show repeated microbial blooms and poorly oxygenated conditions for millions of years after forest collapse, demonstrating how damaged landscapes can continue producing harmful conditions that delay broader recovery. Chance also matters. A species may survive because one population happens to live in a protected valley. A storm may miss one island and strike another. A generalist may reach a newly open habitat before its competitors. A mutation useful under the new conditions may appear in one lineage and not another. This makes the distant future impossible to name species by species. We can predict broad tendencies, not a caste list. If current pressures continue, the near future would probably contain many organisms already familiar with human environments. Small mammals, adaptable birds, insects, fungi, microbes, weeds, crop-associated species and organisms transported through trade would remain important. Domestic animals and cultivated plants would continue wherever human societies maintained them. Some threatened species would survive in protected areas, breeding programs, seed banks, aquariums, gardens, and isolated wild refuges. Their long-term future would depend on whether those places were large, connected, and stable enough to support self-sustaining populations. A seed bank can preserve genetic material, but it cannot preserve an entire forest interaction. A frozen tissue sample does not store learned migration routes. A captive animal does not automatically retain the ability to avoid predators, find seasonal food, or choose a suitable nest site in a changed landscape. Technology could help prevent extinctions and restore some populations. Genetic tools may support breeding, identify hidden diversity, or improve resistance to disease. Assisted migration may move species towards suitable climates. Reproductive technologies may help animals with very small populations. These methods would remain supplements to habitat and threat reduction. Producing more individuals is of limited use if every safe place for them continues disappearing. A laboratory can assist a species. It cannot provide a functioning continent in a storage cabinet. People often bring up de-extinction when discussing a future after mass loss. The idea is that DNA, cloning, or genetic engineering might recreate vanished organisms. Even where enough genetic material exists, the result would not restore the original population, learned behavior, microbiome, habitat, or ecological community. It could create an organism resembling the extinct one, which is scientifically remarkable, but it would not make extinction harmless or reversible. For most species, usable genetic material will not remain. Small insects, fungi, plants, and marine organisms can disappear without preserved cells. No future technology can reconstruct a species whose biology was never recorded. The most realistic way to influence life after a mass extinction is therefore to prevent as much extinction as possible before the survivors are chosen by crisis conditions. This does not require keeping every ecosystem unchanged forever. Nature changes continuously. Coastlines move, rivers shift, species migrate, and evolution produces new forms. Conservation is not an attempt to freeze Earth at one date. The goal is to keep change slow enough, habitats connected enough, and populations large enough for living systems to respond without losing most of their options. Genetic diversity is an option. A migration route is an option. Several separate populations are options. A varied food web gives predators and prey alternatives. A species living across many habitats has more chances to survive a local disaster. Mass extinction removes options faster than evolution can replace them. On a human calendar, the recovery period would feel almost motionless. The high-productivity ecosystem established in the Chicxulub crater within 30,000 years counts as rapid recovery in geology. 30,000 years ago, humans were living in the late Pleistocene. No city, government, company, or conservation program can make ordinary plans across that interval. A recovery lasting 8 million years is even harder to place in human terms. 8 million years ago, our own lineage had not yet produced the genus Homo. Waiting for evolution to repair a mass extinction is not a practical environmental policy. It is a statement that the planet will eventually contain diverse life again, after everything familiar has been given enough time to become ancient rock. Humanity might survive a severe biodiversity crisis. A mass extinction is not automatically the extinction of humans. We are widespread, adaptable, technologically capable, and able to eat a broad range of foods. Those are useful survival traits. Survival is not the same as living well. Agriculture depends on soils, water, climate, pollination, pest control, and genetic diversity. Cities depend on reliable food and water arriving from other places. Fisheries depend on functioning marine food webs. Extreme environmental change can increase conflict, displacement, disease risk, and economic instability. It would be a peculiar success to remain alive on a planet where much of the biological support for civilization had been damaged. Humans can replace some ecological functions with machines, chemicals, infrastructure, and labour. Doing so is usually expensive, incomplete, and unevenly available. The better outcome is not to prove that our species can tolerate the world after a mass extinction. It is to avoid carrying out the experiment. So what would life become next? At first, it would be whatever survived. The organisms that could use damaged habitats, reproduce under unstable conditions, and find food in simplified ecosystems would spread. Some places would be dominated by microbes, algae, fungi, small animals, pioneer plants, and generalists. Other places would retain complex refuges containing remnants of older ecosystems. Over thousands of years, communities would reorganize. Over millions, surviving lineages would diversify. New predators, herbivores, pollinators, reef builders, and forest plants would appear. They would not recreate the present. They would produce another version of Earth's biosphere from the limited set of lineages that remained. The exact result depends on what disappears beforehand. Every species saved now becomes one more possible ancestor, competitor, pollinator, predator, prey animal, seed disperser, habitat builder, or evolutionary starting point in the future. That is why the question, how long until the next mass extinction, cannot be answered only with a date. The meaningful measurement is how much biological possibility remains each year, and whether human action is increasing or reducing it. We began by asking which living thing you would notice first if it disappeared from the place where you live. Perhaps you thought of a bird outside your window, insects in summer, a tree along a familiar road, or an animal you've never seen in the wild but are glad still exists. That species does not need to be the last of its kind before its decline matters. It does not need to become a fossil before it becomes part of Earth's history. It needs enough habitat, enough individuals, and enough time to continue. The 5 previous mass extinctions show that life is persistent. They also show that persistence is not protection. Life continued, but entire worlds of species and relationships ended. Recovery produced new ecosystems only after intervals far longer than human civilization has existed. The current crisis is not complete, and its final scale is not fixed. That is the most useful fact to carry away. Some species are already gone, many others are declining. Yet populations have recovered when pressure was removed, habitats were restored, and conservation continued long enough to work. There is still time to influence what survives, which functions remain, and how difficult the next centuries become. Not unlimited time, and not one shared countdown for every species, but real time in which causes can be reduced and outcomes can change. And now, as we reach the end of this long look at extinction, take a moment to notice what is still living around you. It may be very quiet where you are, but quiet is not the same as empty. A tree may be moving water through its leaves. Fungi may be breaking down wood. Insects may be resting in soil. Birds and mammals may be waiting for daylight. If this slow journey through Earth's past and possible future helped you understand the question a little better, you can like the video, subscribe, and share it with someone who might enjoy calm science at the end of the day. The next mass extinction is not an appointment we're waiting to attend. It is a risk measured through thousands of populations and millions of choices. What happens next will be determined partly by environmental forces, partly by chance, and partly by what people decide to protect while protection is still possible. For tonight, there is nothing more you need to calculate. The planet is still full of living things, and many of them still have a future that can be changed. Sleep well, wherever you are listening from,